The board of 5th Hybrid International Conference on Molecular Modeling and Spectroscopy is celebrating with Professor Amr Mohammed Abdelghani, as her appointed the DSc in Experimental Physics, from Mansour University 2023. This overview is going to make short biography for Professor Abdelghani. First it will be an overview in Arabic then in English.
هنا صدقت حلمي وبدأت قالوا عني زمان موهوم بيضيع وقت لا يوم سلمت ولا استسلمت ووقعت وقمت وامبارح غير دلوقت هنا قلبي من جوا ده اللي بيديني القوة في طريقي مشيت واديني ثبت من هنا قررت بعد ما فكرت امشي عالصعب وعرفت انا لما كبرت ان اللي تفكر في ازمة كان خوفك منه ملوش لازمة واديك عالصعب قدرت واللي زمان ضحكوا عليك شوفهم دلوقتي بيداروا كسوفهم وراهنوا عليك تخسروا كسبت انا ابن مصر انا ضد الكسر انا رافع راسي تملي لفوق طول العمر انا ابن مصر قلبي نسر ما اقدرش اعيش ابدا يوم غير وانا حر شفت يا زمان ومواقف ملهاش عد بس الصدمات بتقوي ومش بتهد شفت يا زمان ومواقف ملهاش عد بس الصدمات بتقوي ومش بتهد انا عندي ثقة بالنفس ومناعة ضد اليأس انا ابن مصر انا ضد الجسر انا رافع راسي تملي لفوق طول العمر انا ابن مصر انا ابن مصر قلبي نسر ما اقدرش اعيش ابدا يوم غير وانا حر انا ابن مصر انا ابن مصر انا ضد الكاس انا ضد الكاس انا رافع راسي تملي لفوق طول العمر انا ابن مصر قلبي نسر ما اقدرش اعيش ابدا يوم غير وانا حر.
Professor Amr Mohammed Abdelghani graduated from the Physics Department, Faculty of Science, Mansoura University in 1992 and was appointed as a demonstrator in the Department of Physics. Obtained his degree of PhD in Experimental Physics from Mansoura University in Egypt in 2010. He was appointed to the position of Professor of Applied Spectroscopy in 2020 at the National Research Center. Worked as a mandate lecturer at Mansoura University, Nile Academy, and Horace Egypt University. Supervised more than 100 master's and 40 PhD theses. He served as administrator of the Physics Division of the Syndicate of the Scientific Profession and vice chairman of the Syndicate of Scientific Professions Mansoura branch. He has more than 234 worldwide research publications including in respected journals with more than 5,730 citations and H-Index 51 Scopus-based and 4 international published books. He was a PI and co-PI member in 20 national and international projects. He was awarded the Incentive Award, 2013, the Pioneer Award by Professor Shihada Farag, 2014, and Professor Kamal Nasser Abdel Noor Award, 2015, at the National Research Center and Professor Amin Lotfi, 2016, at the Academy of Scientific Research. He was a member of the National Committee of Physics during the period 2018 to 2021 and was elected as chairman of the Egyptian National Committee of Physics during the period 2022 to 2025. Recently he was awarded a degree of Doctor of Science in Experimental Physics in 2023. He is conference co-chairpersons of 5th Hybrid International Conference on Molecular Modeling and Spectroscopy. The board of the conference is delegated to celebrate with him his scientific achievements. More details about Professor Abdelghani could be found at both ResearchGate and Scopus as in the following web pages.
Bismillahirrahmanirrahim. Esselamu Aleyküm ve Rahmetullah. Dear participants, salam from Turkey. I'm very happy to be again with all of you, all of my friends at Egypt, and also other friends from different countries from the world. This conference is very fantastic for me, and it is very important for me and my research group wait every year for this conference, and Alhamdulillah we are again at this conference. Maybe next times we want to be at Cairo with face-to-face, inshallah. And I want to start to my speech with some thanks. Especially, I want to thank my dear brother and my dear sister, Dr. Mithat and Dr. Hanan, and also thanks to organization committee for inviting me to be a part of this conference. Thank you very much. It is very important for me. I will talk about green sample preparation techniques, especially separation and pre-concentration procedures, some explanations about them for the determination of trace and inorganic species. And I want to start firstly trace environmental pollutants. Maybe we can divide trace environmental pollutants two parts. One part is organic pollutants, other parts is heavy metal pollutions. Both of them, even at trace levels, very big problem for human beings and environment. For example, organic contaminants as including pesticides, microbial pollutants, dyes, pharmaceuticals, persistence, organic pollutants, and others are big problematic roles in environment. Also, they can cause different kinds of problems in human life, including anemia, cardiovascular disorders, memory disruptions, and others and others. And also, another parts of trace pollutants are trace metals, trace elements, including lead, arsenic, chromium, copper. At even trace levels, these kinds of metals, especially lead, arsenic, chromium, and cadmium, in environmental samples is a big problem in human beings. For example, lead could be sources cancer, renal, kidney diseases, and others, and chromium sources, chromium as problematic on diarrhea, headache, carcinogenic problems, and etc. Due to these points, their determination in environmental samples are very important. But in their determination at trace level, both of organic and inorganic constituents, the main problem is we have two main problems. One is matrix effects. Second is lower element, lower analyte concentration. Instrumental techniques sometimes could not determine these kinds of organic and inorganic constituents due to matrix contents of real samples at higher level and also lower level of analysis than the instrumental detection limits of your instrumental system. To solve these kinds of problems in analytical chemistry, in atomic chemistry, in organic determinations, we should use separation and preconcentration techniques, especially to overcome matrix defects. Separation is necessary, and also lower analyte levels, to solve lower analyte levels problems, preconcentration, or we call it as enrichment, is necessary. Due to these points, sample preparation and especially extraction techniques and is very, very important part of in the chemistry studies. Different kinds of solutions are possible, but cleanup, evaporation, treatments. But the main to solve these kinds of problems, extraction is the one of the main points. And also my groups are very important place about these kinds of studies in chemistry. And also at the approximately 60 years, 70 years, scientists could use these kinds of methodologies. But after 19th, a new world, green chemistry, green analytical chemistry, we heard. And after 2000s, we used these kinds of methodology, and also it related with extraction techniques for trace metal, trace inorganic and organic analysis. The timetable as shown is here, and green chemistry is going on, and also extraction, new achievements on the extraction technique are going on. Both of them is very important. In the green chemistry, in the green analytical chemistry, there are 12 new rules of green chemistry. For example, you should prevent waste, and also your methodology should use less hazardous chemical materials, safer chemical materials and products you should use, and design chemicals and products to degrade after use, and blah blah. Both 12, all of 12 rules of green chemistry is a very important in nowadays in extraction techniques. Due to these points, especially for the separation and preconcentration of inorganic and organic analytes, micro extraction techniques are placed to other techniques because they are green techniques, and in liquid micro extraction, in solid phase macro extraction techniques, you can use the rules, all the rules of green chemistry. Due to these points, our research group approximately over than 10 years focused for the green chemistry rules, and green micro extraction techniques. Again, minimization and miniaturization your system gives us to go on green analytical chemistry rules. Due to these points, different kinds of solid phase micro extraction techniques, liquid phase micro extraction techniques, including solidified micro extraction techniques, and hollow fiber liquid phase micro extraction techniques and others, and also different kinds of solid phase extraction techniques were used for these kinds of studies, because energy efficient, miniaturized instruments, safe non-toxic solvents, minimized waste, low sample and solvent volumes, and fast analysis. It was very important for analytical chemists and all of the scientists, and also low operation cost our techniques has. Due to these kinds of green solvents, green sample preparation, and green separation techniques combined with different kinds of instrumental techniques for organic analyze, liquid chromatography, for it's also gas chromatography and others, for inorganic analyze, especially for metals, to use atomic absorption, ICPMS, and others. Also, these kinds of techniques could be solid phase extraction techniques, micro level, as called micro solid phase extraction, and liquid phase extraction is possible and also some examples could be given membrane assisted extraction. You can see here different kinds of methodology for liquid micro extraction and solid phase macro extractions, and different kinds of strategies are possible. At the continuance of my slides, I will give some examples performed in my laboratory about them. For example, new kinds of extraction solvents, new green extraction solvents at this area, very attractive, and I will talk especially hypothetic solvents, ionic liquids, supramolecular solvents, and also in solid phase micro extraction studies, dispersive solid phase micro extraction, thin film solid phase micro extraction, magnetic solid phase extraction, and others could be used for to solve these two problems by the scientists around the world. Again, you can see another scheme for the solid phase extraction studies, especially solid phase micro extraction studies. There's many kinds of possibilities you can use an application with different kinds of materials you can use, and especially we call these kinds of materials in solid phase extraction as adsorbent. You can use different kinds of new adsorbents for the separation and preconcentration of your analytes. And also here you can see some again some application of these kinds of methodology, especially graphene oxide, very attractive materials, and also nanodiamond and diamonds, and some kinds of carbon nanotubes, including single-walled carbon nanotubes, multi-volt carbon nanotubes, and metal organic frameworks and covalent organic frameworks could be used for the micro extraction, solid phase macro extraction of trace organic and inorganic analysis for the separation and pre-concentration. Again, we can look at methodology. Your sample, aqueous sample in this tube, and you should add your adsorbent at approximately milligram levels to this solution. After some procedures, including by using sonicator or vortex and others, the adsorption process, after adsorption process, you can use separation process by using centrifuge, or if your adsorbent has magnetic properties, you can use on separation by a magnet. After separation of adsorbent, your analyte adsorbent on the surface of your adsorbent, you can, after discharging aqueous solution, then you add eluent solution for the desorption of adsorbent analyte on the surface of your adsorbent. Then using some methodology, again, sonification or vortexing. Then after separation of your adsorbent, your analysis on the eluent solution, you can analyze it according to its properties. What is its properties? If it is organic materials, you can use HPLC, ionic chromatography, GC, gas chromatography. If it was metals, you can use atomic absorption spectrometry, you can use inductive coupled plasma spectroscopy, and etc. Different kinds of instruments we can use in the determination step. Nowadays, many new and different adsorption materials are very popular. For example, layered double hydroxides, metal oxides, metal organic frameworks, carbon-based materials, biopolymers, bioadsorbents, and others for the separation and pre-concentration of trace analytes. Our research group are focused nowadays combination of metal organic frameworks and layered doubled hydroxides. I will give some example about them. Also, you can add magnetic properties to your adsorbent for the easy separation of your analytes or easy separation of your adsorbents. Due to this points, magnetit is one of the important materials to give the magnetic properties your adsorbents, and you can use different possibilities by using combination of magnetit and carbon-based materials, magnetized ionic liquids, and magnetized layered double hydroxides, and others. So I want to give, after this explanation, I want to give some example studies from Soilak Research Group about solid phase extraction, solid phase, microsolid phase extraction, and liquid phase extraction. These are our new hot works For example, here we used metal organic frameworks after combination with magnetic to or magnetic properties, to give magnetic properties of our adsorbents. We synthesize these materials by using hydrothermal synthesis units and by the aid of pressure and temperature. After synthesize these materials, we should characterize our materials by using different kinds of methodology, including heat emission, scanning electron microscope, X-ray diffraction, infrared spectroscopy. Here you can use, you can see some characterization steps. After characterization, we use these materials for the solid phase extraction of cadmium at trace levels prior to its flame atomic absorption spectrometric determination. All analytical points of all analytical points are optimized for each studies. Another different methodology for one of the pesticides, which is called as atrazine, for the atrazine determination by using high performance liquid chromatography system. We used magnetic multivolt carbon nanotubes for this purpose, because of everyone knows that multivolt carbon nanotubes has very attractive absorption properties, including high surface area and high porous structure. And due to these properties, we use these materials. We characterize these materials by using again the characterization techniques, and we use these materials for the solid phase extraction of solid phase macro extraction of atrazine at natural water samples, and we find higher level of atrazine in analyzed samples from the environment. This shown that the usage of atrazine in especially agricultural area is big problem. Another example for these kinds of materials we called nanoflower. Their shape, their scanning electron microscope shape is like a flower as shown here, but not blue and discolored. The materials is this black colored, but it's like these kinds of flowers. Due to these points, we called them as nanoflower. We can use these kinds of nanoflowers and on the separation and pre-concentration techniques, on the micro solid phase extraction techniques. Here is an example for these kinds of work. Silver modified zinc oxide for the nanoflowers for the solid phase extraction of lead from food and water samples. Again, after synthesize these materials by using hydrothermal synthesis unit, the material was characterized by using some FTER and XRD and others. Then the materials used for the solid phase extraction of lead. Again, here is the same image of this materials. It's shaped like a flower. The methodology is here, absorption of our analytes at pH 4. We have quantitative recoveries for our analytes at pH 4 after elution with diluted acids. We could determine our analytes by flame atomic absorption spectrometry. Another example, as shown here, is the determination of atrazine with different materials: activated carbon cloth, nickel cobaltate, and magnetic combination. And we can easily determine, we can easily preconcentrate our analytes by using these materials, and the determination of atrazine in different kinds of environmental samples, including water and food samples, were performed. Another example in our research group, magnetic solid phase extraction of patent blue dye. As pointed in the beginning of my speech, dyes are a big problem in the environment, especially textile dyes and food dyes are a big problem, and their determination is very important. And this study is related of patent blue 5 in different food and different shrubs, water, and artificial sweet samples. It's by application was very successful performing these kinds of materials. And another example, as shown here, one of my Sudanese students performed this study. Both two are Sudanese, and they have loofah from their country, and we combined these materials by adding titanium oxide and magnetic properties. After characterization, these materials, we use these materials for the solid phase extraction of lead at trace levels, different kinds of food and water samples. Also, we have some review studies for these micro extraction studies. Here you can see samples of micro extraction of pesticides from environmental samples and food samples. I can send, if anyone wants these studies, these papers, I can send them by email. Another review on this area, the micro extraction technique based on new trends in food analysis, because the determination of these kinds of organic and inorganic trace metals in food samples are very important. This review is related with this work. Another liquid phase micro extraction studies for this area. Again, determination of a dye, Sudan 3 dye in food samples by using supramolecular solvents. They are very new and very green solvents, and the determination of Sudan 3 were performed successfully in different kinds of food samples in this study. Another examples here switchable solvents. They are called as smart solvents. You can arrange the structure, the properties of your solvents by adding ammonia or sodium hydroxide, and after addition you can change the properties of solvents, and due to this point they called smart solvents, and we use these materials on the determination of erythrocin in food and other samples by spectrophotometric determination. And also another important new green solvents are dipotectic solvents. They are very important at this area. They are very attractive for the micro extraction studies, especially they are very attractive for the micro extraction of bioactive materials from plant samples. And also we use these materials for the separation and preconcentration of some metal ions in oil samples from our country. We have very successful results for this by using for these solvents, these kinds of solvents, and we will continue these kinds of studies in the next. Another example, again, dye sample, sunset yellow. It says it is an important dye in food and pharmaceutical products. We have a new methodology for the dispersive liquid phase micro extraction of sunset yellow. In this work, we applied our methodology various drugs, vitamins, beverage, foods, and environmental samples. Another example here is determination of rhodamine B. It's again important dye in food and food samples, and to determination their level in food and water samples is very important. We use in this study we use a new dipotectic solvents, quasi-hydrophobic dipotectic solvents for the separation and preconcentration of rhodamine B in water and food samples. Now in future, we are going to use quantum dots in solid phase extraction and solid phase micro extraction. Our studies continue with metal organic frameworks and their nanocomposites, and also covalent organic frameworks we are going on, and nano extraction and solid phase nano extraction studies we are aimed in our next stage. And also we have a new area about nano engines, nano membranes, and microfactories for the especially drug delivery studies. In a short time, we will publish, inshallah, some papers about especially nano engines and micro factories for the drug delivery studies. And also, at the end of my talk, I want to thank Turkish Academy of Science. I am one of the members of this academy. I want to thank to them for their financial support, and I want to thank to TÜBİTAK, which is Turkish Science Council. Also, their financial supports are very important for us. And also, I want to thank my university, RGS University, for all things. We are very happy at my university, and I want to thank my research group members. They are without them, I cannot do anything I want to thank all of them. And we call that Soylak Araştırma Grubu, means Soylak Research Group. And also there are some, there is some photos from my city and my research group. I want to invite all of you to my city, especially Dr. Ibrahim, Dr. Mithat Ibrahim and Dr. Hanan. We are waiting you for Kayseri, your Kayseri visit. And thank you for your attention. Thank you for your listening. Teşekkür. Thank you very much.
Good afternoon, everyone. Honorable Conference Administration, Chairperson and Co-Chairpersons, Honorary Chairperson, Professor Mithat Ibrahim, dear participants, ladies and gentlemen, it's my great honor to attend the 5th Hybrid International Conference on Molecular Modeling and Spectroscopy and present my talk entitled Photo-Induced Netosis. I apologize for some hoarseness in my voice. The topic of my talk is devoted to one of the examples of the successful application of molecular vibrational spectroscopy, namely Raman spectroscopy in life sciences. The topic is netosis, which is one of the programmed cell death. I believe there is a sense to give some general brief information on neutrophils, which are actually the largest population of leukocytes. It's important to note that when the host organism is infected, neutrophils immediately migrate from the bloodstream to the site of infection, providing a first line of defense against bacterial, fungal, and viral infections and pathogens. In this case, the main protective mechanism are phagocytosis. This one, you can see here a video with neutrophil cell catching the pathogen, chromatin decondensation, and ultimately at the last stage, the formation of neutrophil extracellular traps, or simply nets. You can see here the schematic illustration of that. And nets are basically modified chromatin decorated by bactericidal proteins of granules, nucleus, and cytoplasm. Presently, two forms of netosis are known: classical or suicidal netosis and the vital netosis. Net formation can be induced by a large variety of physiological stimuli, such as bacteria, fungi, viruses, antibodies, cytokines, and others, and also by pharmacological stimuli such as formol myristrate acetate, or simply PMA. And the third position is the light photo-induced netosis. Presently, the majority of publication on this topic is devoted to the study of netosis under either physiological or pharmacological stimulation. However, in the recent years, the very first works on photoactivated netosis under the influence of UV radiation have appeared as well. Just a few words about the isolation of primary human neutrophils. Peripheral blood was collected from healthy volunteers according to their written consent and following the recommendation of the Ethical Committee of Moscow State University. Neutrophils were isolated according to the protocol. You can see it here. I will not go into the details. So they were isolated by centrifugation in double density gradient using fecal HIPAC. And as a result, fecal falls between neutrophils here and mononuclear cells, which allows you to collect the neutrophils afterwards. For the further activation of neutrophils, we used 96 well plates. We choose Raman spectroscopy as our molecular spectroscopy tool, and we started our first experiments on netosis by deciding to test the sensitivity of our instrument to possibly detect the prenetotic state even before the formation of neutrophil traps. In these experiments, we used PMA and bacteria for activation. In order to realize this test, one must understand the mechanism and the signaling pathways of both prenetotic and netotic cell formation. In fact, this is a rather complicated process, which is schematically demonstrated here on the left figure. And there are several mechanisms and pathways of nets formation depending on various factors. However, on the right part of the slide, you can see a list of the main components and organelles that make up a neutrophil cell and involved in netosis. Some of them, like NADPhoxidase, a membrane-bound enzyme complex, one of the major sources of cellular ROS, peptidyl arginine deaminase known as PAD4, and mitochondria play a key role in the mechanism of net formation. So 3 years ago, we began our research on netosis by targeting the signaling pathway through the PAD4. The reason is the following. It's known that upon activation of neutrophils, radicals stimulate PAD4 to migrate from the cytoplasm to nucleus membrane and citrullinate arginine, resulting in loosening, weakening of chromatin from histone, and thus its subsequent decondition. If so, we probe to apply sensitive Raman spectroscopy to detect the kinetics of citrulline appearance in the spectrum before nets are formed, which typically occurs after 2-3 hours of pharmacological or physiological activation. So for this, we used our multimodal optical platform based on highly sensitive Raman microspectrometer cars, which has a large number of functional options. But I will not go into details here because I don't have time for that. Here are the main results of that part of the study. Neutrophils were activated with T stimuli, with PMA, and with bacteria, and the corresponding Raman spectra obtained are demonstrated here. You can see that the peak with the Raman shift of about 166 centimeters corresponds to the citrulline lattice vibrational mode, and it's practically absent in inactivated neutrophils here and here, and appears only after the stimulation. We can assume that it is associated with the accumulation of citrulline in the peak in the cell. We revealed the evaluation growth of citrulline peak within 30-40 minutes. You can see the central Raman spectrum. After the beginning of the inflammatory process, which can be definitely classified as an early diagnosis of netosis, because usually it takes 2 and more hours to form the nets in the system. So having obtained the first encouraging results on the investigation of netosis by molecular spectroscopy, in particular with Raman spectroscopy, we then moved to a little studied form of netosis activation, namely photostimulated netosis. The study of photoinduced netosis, to our opinion, is of great interest, not only from the fundamental point of view, but also from the point of view of protecting the human body From an uncontrolled dose of solar radiation in a wide spectral range from UV, especially UVA, to near-infrared radiation. This also applies to various sources of artificial light, which are widely used for medical purposes. To make the correct choice of wavelengths of light sources used in an experiment, it is necessary to understand what primary photoacceptors exist in a particular cell. As to induce a photobiological effect, any radiation must be absorbed by a functional chromophore molecule located in some key cell structure capable of influencing its activity. So it's known that the redox chains are an example of such key structures. In neutrophil granulocytes, components of the mitochondrial respiratory chain, in particular cytochrome C oxidase, and the membrane-bound flavohemoprotein cytochrome B885 can be considered as effective photoacceptors and transducers of the photosignal. So we put forward a hypothesis that these two cytochromes, C and B885, could play a role of primary photoacceptors in photoinduced netosis. So the selected neutrophils were irradiated with V-late wavelength switchable LED sources at the following 5 wavelengths. First of all, UV 365 nanometer, then comes blue one, green one, orange, and the red LED source. The choice of these LEDs, with the exception of the 656 red one, was adapted to the absorption spectra bands of the two cytochromes mentioned above. The cells were irradiated at room temperature in a selected dose-dependent manner, with the doses of 4, 16, and 35 joule per centimeter square. And to confirm our hypothesis on photoacceptors, primary photoacceptors, we used 3 targeted inhibitors of NAT oxidase, PAD4, and mitochondria. Since activation of neutrophils leads to oxidative stress inside the cell, in particular to the generation of various radicals such as superoxide, hypochloric acid, hydrogen peroxide etc., we started our Raman experiments targeted on recording these radicals. All the Raman measurements were performed in the mapping mode during the first 10-15 minutes after neutrophil activation by LEDs. You can see here the micrograph here of the neutrophil cells. In the center is the Raman map, and on the right you can see the Raman spectrum with one very sharp line, intense sharp line, which belongs to the vibration of the hydrogen peroxide. The purple pixels on the map are also positive here, although the intensity of the signal is not so strong here. This particular map was obtained with the exposed radiation at 625 nanometer and a dose of 32 joule. However, very similar results were registered at the other wavelengths as well. Another radical which we detected in our experiments is the hypochloric acid. You can see here the corresponding Raman spectrum. This peak at 732 inverse centimeters is assigned to characteristic Raman frequency of hypochloric acid, detected at the exposure of neutrophil cells with LED at 405 nanometers. The vibrational frequency here originates from the oxygen-chlorine stretch mode. So for the first time, the Raman spectroscopy was applied to detect the radicals generating during the oxidative burst upon irradiation of cells during the first 15 20 minutes after simulation of neutrophils. We also returned to the search of citrulline spectrum, as it was done in our earlier experiments with pharmacological stimulation of cells, but this time with improved filters operating in the low-frequency region of the Raman spectrum. We managed to register not one peak as it was in our first works. You see the spectrum on the right part of the slide, but 3 vibrational peaks of the citrulline lattice. Due to the high quality of so-called Bragg filters, the Ramanship of the central peak in the spectrum was measured this time more precisely. We also applied fluorescence microscopy to identify and quantify netotic and vital cells exposed to both UV and visible light in a dose-dependent manner. The corresponding results are shown on the slide for negative control or non-irradiated cells, for positive control PMA, and for 2 wavelengths, 365 and 625 nanometers at 3 different doses. And we can see here clearly that the higher dose, the higher is the output of netotic cells at the both wavelengths. The full-scale results of fluorescence microscopy are presented on this slide for all 5 irradiation wavelengths at a dose of 32 joule per centimeter square. Using inhibitory analysis, we established that photoinduced netosis proceeds through NAT oxidase and PAD4. Thus, the hypothesis on cytochrome B558 as a primary photoacceptor was at least preliminarily confirmed. Additionally, this concept was also supported by the low level of netotic cells yield at an irradiation wavelength of 656 nanometer, this was the last one here in the red, which is absent in the peaks of the absorption spectrum of cytochromes. Since we don't have a specific mitochondrial inhibitor at the height of these studies, we performed this experiment a bit later. Here are those results, and we can see from this comparative diagram that mitochondrial inhibitor mitotempo dramatically suppressed the formation of NETs. Dear colleagues, in concluding my presentation, let me once more demonstrate the schematic of photoinduced netosis activated at the exposure of UVA and visible light. We have identified 3 signaling pathways leading to this phenomenon through NAT oxidase, through PAD4, and mitochondria. Fluorescent analysis showed that netosis appears to form under the influence of both UV and visible light, although different wavelengths result in different degrees of netotic cell release. All these factors, to our opinion, are certainly important. However, it should be considered that human skin is simultaneously exposed to a wide spectrum of sunlight, including the infrared one. This is in our plans, and those of you who are interested in this topic are welcome to cooperate with our group in the future. Finally, let me demonstrate the key findings on photoinduced netosis. Really, for the first time, netosis was detected not only under the action of UV, but also visible radiation at 4 wavelengths from blue to red. Spectral radical biomarkers of photoinduced netosis in its initial stage of oxidative stress have been registered for the first time by Raman spectroscopy. A hypothesis was put forward and proved about the primary photoacceptors of photoinduced netosis. Understanding the molecular mechanism of various forms of netosis is important for regulating unwanted net formation. But this is a topic for a special presentation. Dear colleagues, the work was carried out over a period of about 3 years and partially, not completely, published in the special issue of ICMMS journal named Raman Spectroscopy for Chemical and Structural Characterization in Biology 2. I completed my talk and thank you very much for your kind attention.
Hello, everyone. First of all, let me thank all the organizers for this conference and for their efforts, to put all this material together. I know it's not an easy task because I have been there. And also, I would like to thank Dr. Midhat Ibrahim for the kind invitation to be part of this conference. It's very, very, from what from what people describe to me, it's a very important, initiative, and I'd like to be supportive of it in the future. For those who know me, they know that I'm a people's person. I like live audiences. When I interact, it's for better interaction during the presentation and so forth. So I will do my best in today's presentation that is recorded for logistical reasons. So I will do my best. As a housekeeping item and also as a disclaimer, I have to say that I have calibrated this talk for graduate students or recent PhD graduates. So apologies for senior colleagues, senior members who may see that some of the material is redundant detail or something that is common knowledge. I just wanted to give that disclaimer before we start. Finally, I'd like just to give a very brief outline. The spirit of the conference is spectroscopy, modeling and spectroscopy. So when Dr. Ibrahim asked me of topic, FRIT came to mind because it's basic spectroscopy, but it has taken the recent decades. It has taken a very different trajectory, and actually it's being used in many areas, and particularly in biology, where it sheds light on interactions within the cells, single cells, as we will see later. So I will start with an introduction and also give the basics, of FRET, basically the physical chemistry behind FRET, and then I will finish with some applications as we move forward. Hopefully I can pack all of that in 30 minutes. So apologies if you see me moving faster for certain slides, because there's a lot of material to cover. Typically, this type of presentation about FRET, I do it, this is actually, I use this, as materials, for my graduate course, macromolecular structure and dynamics. And we typically cover this over all the material over, a couple of weeks to really go into details with the physical, chemistry and, all the, the stuff and how it is used. So I will do my best to pack everything in 30 minutes. But for the audience, particularly for the junior members, know that I will skip a lot of details. So hold on. So let's go for it. So let's start with the spatial resolution problem. Obviously, in spectroscopy, we want to resolve things. We want to measure things in finite, distances, and also in time. So we want to resolve things in space and in time. Let's put the time aside and talk about space. When you talk about the ruler that you see up there, here, measures things, from the tissue level all the way to the molecular level, and gives you some sense of the distances that we are talking about, from the millimeter scale all the way to the nanometer scale. Things that happen within the cells are actually at, the molecular level, of course, protein-protein interactions, and these happen at the nanometer scale. So obviously you can think about a microscope to look at things. When you have your magnetic, the microscope, you can look at, for example, cells, within cells. But can you look at things, at events that are happening within cells? You are limited in microscopy using the microscope. You are limited by the wavelength of the light that you are using. This is what we call the diffraction limit, the Rayleigh limit, the Abbe limit. So this, there's a lot of details, of course, that I'm skipping, but essentially the light that you are using limits basically the resolution that you can have, which is, give or take about half the wavelength. If you are using a 550 nanometer light, it's about 225 nanometers. That's the limit of resolution that you can have given the diffraction limit of the microscopes. So when you are using this type of microscopy in immunofluorescence, when you label things with fluorescent proteins or fluorescent material, what you are looking here, yes, you are seeing some details of interactions of different colors. So you can think that, well, the two are actually interacting. But what you have in this picture is actually just a crowded picture that you see when the analogy that I give is that of an auditorium where you have a lot of students and you shine light on them. You see all of them in that auditorium. So this picture, what it tells you, that two students A and B are in the auditorium. It doesn't give you any details about their interactions, whether they know each other or don't know each other, and so forth. What we want to know, we want to have a method that gives us an insight into the interaction between students. Does this student A know student B or student C? Do they interact? What type of interactions? Because this type of interaction between protein interaction is really what drives function at the biological, in biology. Biology is driven by protein interaction for the most part. And knowing this type of interaction will give you insight into cellular function. And that's really the target. And this is where Fred comes. We will introduce this technique to see that it actually introduces a yardstick that gives you information at this nanometer scale. So it is really a nanoscale yardstick, as we will see. So let's move forward. So let's start with the basics, and the place that I typically would like to start when I talk about fluorescence basics is the Jablonski-Perrin diagram. So it is a schematic presentation that tells you basically the various transition between the electronic levels, and then the relaxation between, of course, higher levels and lower levels. The excitation of molecules at the ground state to higher excited states, that's what we call absorption, of course. Sometimes you have this internal conversion where you have coupling between vibrational levels of higher excited states to lower ones, and you have this relaxation, that way. You can have, obviously, at a longer wavelength, this emission of light that is labeled as, fluorescence. Occasionally, you have this intersystem crossing that gives you this long-lived triplet state that also emits light, and that type of light is, labeled as phosphorescence. So the simplified diagram is what you see here. Obviously, absorption of light from a ground state to a higher excited state, various levels, this internal conversion, and ultimately you have emissions, at a longer wavelength. So excitation and then emission. So what is FRET? What is FRET? FRET is an acronym. It stands for Fluorescence Resonance Energy Transfer. And early papers, you find even now, some papers call it the Forster Resonance Energy Transfer. What it is a process by which a donor fluorophore, let's call it D, is in an excited state, transfers the excitation to a nearby chromophore that we call the acceptor. So you have a donor fluorophore and an acceptor fluorophore. You excite the donor, but instead of getting the emission from the donor, that emission, that energy is transferred to the acceptor, and the acceptor emits light. So it is, and this is very important, particularly for our junior colleagues here, it is a non-radiative process. This is not something where, for example, the acceptor gets the excitation and emits light, and that emitted light is the one that is transferred to the donor. That's not the case. This is an internal transfer of the excitation between the donor and the acceptor, and what you get in one step, what you get is the emission of the acceptor. So FRET as a phenomenon is not new. I mean, early papers date back to 1922, for example, in this paper that you see here, where Carrier and Frank reported that irradiation of mixtures of mercury and thallium, where the light is absorbed by mercury and not by thallium, actually results in the emission of both. So somehow one mercury in this case acts as an antenna for the excitation, for the emission of thallium. So how can you use this method to study biophysical interactions, for example? You can imagine, for example, if you have this string of, it can be a protein or polymer. In this particular case, it's a protein. For example, we label one end, you label it with a donor, and then you label the other end with the acceptor. And then let's say, for example, we're studying the folding of the protein. That folding will bring the donor and the acceptor to the right distance for FRET to occur. And that occurrence of FRET will be a measurement of the folding. So this is one way to look at it. Another example where FRET really comes in handy is with, for the interaction of membrane-bound proteins in cells, in live cells. We can imagine, for example, many of you know that biological processes at the membrane are driven by recruitment of various factors, proteins. For example, the recruitment of this protein here at the membrane may induce the interaction of another between this protein that are on the other side of the cell, in the cytosol of the membrane, the cytosol side, where now if this protein is properly labeled with a donor and you excite that donor gets closer to the acceptor, and basically the emission from the acceptor is a signal for FRET, and that signal tells you that the two proteins are now interacting. So again, this FRET can be used to monitor this type of cellular processes. So it is essentially a quantum mechanical ruler. It's a yardstick, a nanoscale yardstick, but based on this quantum mechanics that basically gives rise to FRET. So what are the observables? The question that comes to mind sometimes, always, sometimes you have false positives, in different systems, particularly in very complex biological places where we will see is that you have that problem. The question is, do you have FRET or you don't have FRET, and how can you discern the two? One way to look at it, there are actually few physical, processes that's based on physical chemistry that tells you that indeed you have FRET or you don't have FRET. The first one is this dynamic quenching of the donor's emission. So you start with the donor. Of course, when you excite the donor, you expect the emission from the donor. But as you add the acceptor, what you see is that the emission from the donor is quenched by the presence of the acceptor. That is a dynamic quenching of the donor's emission, and that is a signal of FRET. The opposite, you have sensitized emission from, of the acceptor's molecule. That is essentially when you have just the acceptor and then you excite at the excitation of the donor, you don't see any emission. But as you add the donor, now you excite the donor emits, transmits light, transmits, the energy to the acceptor, and you see this emission from the acceptor. So the sensitized emission of the acceptor molecule in the is actually a signal of FRET. Photobleaching is also another signal that but is typical. It's a process that is typical in, when you excite molecules, continuously. For example, ultimately you may have, photobleaching. So essentially, it's basically the disruption of the processes that lead to fluorescence. So now, when you have a threat, essentially you continue exciting the donor, but in the presence of the acceptor, that photobleaching rate is actually decreased, is reduced. That is a signal that actually the threat is happening because of that internal transfer of the energy between the donor and the acceptor. Likewise, the opposite is what you have is donor-triggered photobleaching of the acceptor, where it is essentially what happens when you have, you are exciting the donor, you are not exciting the acceptor, right? But in the presence of the acceptor and the donor, what you have is you have emission from the acceptor, right? So this particular process here, essentially it's as if you are exciting the acceptor, continuously, and that leads to the photobleaching of the acceptor. So this donor-triggered photobleaching of the acceptor is a signal that threat is happening. So all of these are things that can be checked to make sure to validate that you have threat, that threat is included. I will pass on this increased anisotropy of the donor fluorescence because probably we will not be able to cover it in time. So let's address the requirements. First of all, the requirements for threat to occur. First of all, you must have what we call the overlap requirement, and that is the emission of the donor has to overlap, or the spectrum of the emission of the donor has to overlap with the excitation of the acceptor, so that the extent of that overlap will decide or will determine the extent of threat of the fluorescence resonance energy transfer between the donor and the acceptor to occur. So I show this here as with this spectra here, where you see this is the cyan fluorescence protein. You see this is the excitation of this fluorophore, and this is the emission in solid line. In the excitation of the yellow fluorescent protein is this with this dotted line here. You see that there is an overlap between the emission of the cyan fluorescent protein and the excitation of the yellow fluorescent protein. So this overlap between the two, the emission of the donor and the excitation of the acceptor, is what determines threat. So that is very important in this spectrum here. The other requirement that you must have is the distance requirement. As we said, I mentioned this earlier, that threat is very sensitive to distance, and this is actually the reason why we are using it, because you can actually measure or you can monitor interactions really at the nanoscale. So the donor and the acceptor must be within 10 to about 100 angstroms. That is between 1 to about 10 nanometers. So essentially, threat is this ruler that can measure interactions within the cell, and that is really beyond the capability of light microscopes. So it is, again, because it is, it depends on the distance between the donor and the acceptor. As you see, this is the threat efficiency. If you were to measure threat efficiency, that is the efficiency of energy transfer between the donor and the acceptor, as you will see, it actually decreases as, as a function of distance here. And so if you normalize everything here between 1 and 0, the 50% threat efficiency corresponds to this very distinctive distance R0 that determines basically the distance required to have 50% efficiency between the donor and the acceptor. So, for example, between the cyan fluorescent protein and the yellow fluorescent protein, that distance or characteristic distance is around 4.9 nanometers. The other requirement that is needed is this lifetime requirement. That is, the fluorescence lifetime of the donor must be long enough for FRET to occur. That is, if the lifetime of the donor is very, very short and does not allow the FRET to occur, this dynamics between the donor and the acceptor to occur, FRET will not be possible. So there is a requirement for the lifetime of the fluorescence, the fluorescence of the donor to be long enough for FRET to occur. So let's continue with the basic relationships. We said earlier that the distance is key in the FRET, in the energy transfer between the donor and the acceptor. And this is basically represented here by this rate of energy transfer in terms of kinetics. You see that it is actually, the rate or the efficiency is dependent on the inverse sixth power of the distance between the donor and the acceptor. So we'll see that actually you can use kinetics, just measuring the efficiency of transfer between the donor and the acceptor. The forced critical distance is the R0, that the characteristic distance between the donor and the acceptor that we determined earlier in the previous slide. That gives you the 50% efficiency of transfer. That's called also the Forster critical distance, or the characteristic distance. It depends on a number of parameters. Of course, the mathematics here seems very complicated, but it's actually very simple because most of these variables are actually constants that are found, in this field. So I'll talk about very briefly about at least three of them. This kappa square. Kappa square is basically measures the orientation, the relative orientation of the donor and the acceptor during that transfer. So it is actually proportional. You have to have the right orientation between the two transition dipoles. The transition dipole of the donor and the transition dipole of the acceptor have to be oriented in such a way to allow that transfer to happen. So the R0 depends on that orientation. It also depends on this J of lambda. This is the integral overlap. And that actually depends on the integral overlap between the donor's emission and the acceptor's excitation. So that's the overlap. And also it depends on this QD, which is the quantum yield. Obviously, if you want to have high FRET, you have to choose a pair that has a high quantum yield. So this efficiency of energy transfer that you see here is expressed in terms of the rate of the transfer divided by the total de-excitation. When you use this equation and you add them, you determine here this efficiency, this normalized efficiency of energy transfer between the donor and the acceptor, and you see that efficiency is actually inversely proportional to the sixth power of the distance. So obviously, this is why distance is very, very key, or the efficiency is sensitive to the distance between the donor and the acceptor. So for the sake of time, I'm going to pass on the concept of polarization for fluorescence, but it is important. It is something that's also used to study biological processes, as we will see later. If we have time, we'll cover. If not, we're going to pass on. You can also use FRET in a number of other uses, like, for example, to study the self-association of proteins. For instance, you can use this efficiency, the ratio between the efficiency between different subunits to determine, for example, the number of subunits in a protein oligomers. This is one of the, of course, it's a little bit complicated, but you can do that. So to summarize, FRET can be used in a number of ways. For example, to probe proximity, which is very important in biological systems, to probe also dynamics of protein-protein dynamics or protein DNA type of interaction. You can also determine the local concentration. We didn't talk about homofret, but it is something that can be used to actually probe local concentration of particular analyte. Also, you can use intramolecular and intermolecular reactions or interactions in macromolecules. Essentially, it is a tool that can measure space and time because of the kinetics. You can also measure space, and also because of the kinetics of transfer, you can measure time. So it's spatial and temporal monitoring of all these interactions, whether it's in biology or other systems. So now we will pass to the application side, and this is really fascinating, as you will see. I will go a little bit faster in certain areas because the details may take time, but you will get the bigger picture. So the area that I will focus on is this green fluorescent proteins or other colors that have been engineered and that work as reporters. When you express them in cells, they can work as reporters for various biological events. So this is really used for a biological understanding, also to visualize, within the cells, various events. So this green fluorescent proteins are actually, are expressed by natural, species like jellyfish, and they have been cloned and genetically modified to express also different proteins with different colors, for fluorescence. So you can express the green fluorescent protein, the cyan green fluorescent protein, the red, and the yellow. So this work is really the fruit of many years in three researchers, Nobel Prize laureates. So a Nobel Prize in 2008 has been awarded to three scientists, Shimomura, Shalfie, and Roger Tsien, for their work over decades for the discovery and the development of green fluorescent proteins as particularly as biological markers. So what is this green fluorescent protein? Well, the crystal structure has been determined for many of them. It is a very interesting structure that looks like a barrel, about 2 nanometers of base by 10, and the inside protects the fluorophore that is really at the heart of this, of the fluorescence that we see. So you see the inside, you have this, parahydroxybenzylidine imidazolidone, or it's essentially a dihydrotyosine imidazolidone, and it is the product of 3 amino acids that is within the sequence that mature over time to give you this structure. So as I said earlier, the crystal structure has been determined for many of them, and then you can understand basically the key factors, this tyrosine, if you're in certain of them, the tyrosine, glycine, and serine that come together to form this fluorophore. This is the maturation reaction that has been studied, very carefully, and then you see that ultimately results in this fluorophore that you see here at the end. This dihydrotyosine imidazolidone. I typically ask students, when we get to this year, to just imagine what is this. What do you see here? And some of them see, I see a beach, I see palm trees, I see water, and all of that. Well, it turns out actually this is very interesting. But what you have here, this is a petri dish with, E. coli that is expressing various colors, these fluorescent proteins with various colors. This has been actually, prepared, and I took it off the website of the lab of Roger Tsien, just to express the idea that the what you can do with these fluorescent protein when you express them in various systems. So essentially, this is not really scientifically relevant, but it tells you the possibilities that you can get with this. So you can express really all these kind of colors, and you can use them for various biological species. Now, the variants, as I said earlier, it started with the green fluorescent protein, but other mutants has been made, and you can actually express various colors: the cyan, the yellow, and others that have been done. Well, these green fluorescent proteins or other colors can be expressed in a variety of systems: bacteria, yeast, plants, mammalian cells. They also tolerate the N and the C terminal fusion, so you can express the gene to have them be placed either within the N terminal or C terminal, and they can function, beautifully. They can be placed, in cytoplasmic, in organelle localizations, so to basically to follow processes, within these targeted places in cells. So as I said, they can be really designed to respond to a variety of cellular events, depending on, and people have been very creative in their genetic design. They can also be expressed in variety of tissues, as I said, different cell and intact organisms. What is also interesting is that there is no known photodynamic toxicity. Basically, when you introduce these systems in live cells, there is no known photodynamic toxicity to the cell organism. Basically, the cell behaves just like normal, not normally. And also they can resolve spatial and temporal events, within the cells. So now we will tackle certain applications. These are generic examples of the particular examples that we will go over in the next few slides. But all of them center around the idea that you can design a system that responds either to a particular analyte, or when there is a conformational change between two motifs, or within the same protein in response to a particular biological event. So you can design a system where these FRET changes can give you an idea or can be used to monitor these biological events. So now let's jump on the first of the three examples that we will go over in this last part of the presentation. And the first one centers around this histone phosphorylation. Histones in general are very important in the cell biology. They control and regulate many events within the cell, and one of them, for example, access to the DNA that wraps around them. Phosphorylation is one of these reactions, and it can be followed. So this system here has been designed to follow this phosphorylation. This is a motif of the histone that has been designed to also express the cyan fluorescent protein on one end and the yellow fluorescent protein on the other. And upon phosphorylation, you see the two fluorescent probes. The cyan gets closer to the yellow for FRET to occur, and upon dephosphorylation, the system can be reversed. So this system has been studied where, for example, and you can follow FRET in real time here, as you see here, upon addition of ATP and the kinase, you have the phosphorylation and FRET signal increases. You can also reverse that system by adding the phosphatase that reverses the phosphorylation. So and then upon adding the phosphatase, the FRET signal decreases. And at the base, what you see here at the base here is the FRET signal change that basically remains at basic level. It doesn't increase when you don't have the ATP or when you don't have the kinase. So this is a validation of this phosphorylation of the histone with this reporter, and can be used, as we will see in the next slide, actually even in live cells. So now we will see the use of that histone phosphorylation as a reporter in live cells. It has been introduced in live HeLa cells here, and during the, as we will see in this movie, during the cell division, you will see increases in FRET. And I want you to pay particular attention at the moment of mitotic phase of the cell division, where the two cells, separate, and we'll see increases in FRET. So let's see in this movie, as we will see here, let me use so that I can point you to see, for example, follow this particular cell here. As it divides, there will be an increase in here. It will be an increase in fret, and the same thing is really happening in other cells, as the movie progresses. So you feel free to move the movie forward and backward and stop it to see this particular, event. So this is a very good example that shows the importance of using these reporters to follow live events, within live cells. So this is a static slide of the previous movie that follows a particular cell during the cell division. And as you see, as time progresses, you see this increase in or maximum increase in fret at the moment of the mitotic phase. In the graph here, it shows basically how you can follow fret signal as a function of time, and you can see that it increases around that time frame here. So around this time where it increases. And what you see in the base level here, as for a particular mutant where that serine, the phosphorylation of that serine has been changed, so that basically there is no phosphorylation, and you see that the reporter basically does not show any fret changes. And this is again another validation that the reporter really is reporting on phosphorylation events for this, during the cell cycle of these HeLa cells. Now we will see a second example, also phosphorylation, but of another protein, ABL. The activity of ABL is very important in cell migration and adhesion, and as you will see, upon the addition of a stimulation factor, this platelet-derived growth factor, you will see that the signal of the fret signal will increase, and it will follow essentially this system here. So let's follow the movie here. So as you will see here, the PDGF, which is the platelet-derived growth factor, has been added, and you see immediately, the fret signal is, increasing, and it propagates, as you will see, it propagates, as time passes. So this is another system in which using fret and this reporter, you can follow very intimate biological events within cells. The third and last example follows the activity of EGF receptor, the phosphorylation of EGF receptor in mouse fibroblasts. So as you will see here in this movie, upon the addition of EGF, let's see here, the addition of EGF, you see the fret signal starts at the periphery and then propagates in the whole cytoplasm. So as you will see here, so we're gonna play the movie again one more time. So you will see here addition of EGF, and after some time, you will see that the fret signal starts, at the periphery of the cell and propagates in the whole cytoplasm. So let's see how it goes here. So let's continue. There you go. So this is another example where now you can design systems to follow, again, biological events within live cells. This is a continuation of the previous example where you can follow fret increases after adding the stimulant and then fret decrease after washing out the stimulus. So you can see here the movie as it progresses for both the addition of the stimulant and then after washing out the stimulant where fret decreases. Now you'll see in this the same example, but now in a different configuration of the experiment where you can add the stimulant to trigger the kinase activity, and immediately you overwhelm the system with a phosphatase that reverses the system. So you will see an attempt for fret to increase, and immediately after adding the phosphatase, the fret is basically decreased. So you'll see the movie here. So here the stimulant, and then the addition of phosphatase, where you see that the signal basically does not increase. Lastly, this is another example where now fret can be used to sense ions in general. In this particular case, these are calcium sensing chameleons, when they are modified with a fret pair, that upon addition of calcium, the two fret pairs are closer to each other for fret signal to increase. So this is another example where you can use fret to follow conformational change of a target protein. So there are other extensions of fret that we really don't have the time to cover them, but I include them in this slide for you to check the literature on them. They are fascinating, and the capabilities that they bring. Fluorescence correlation spectroscopy is one of them, and it can be used, brings the capability to follow the dynamics within a particular volume of a system. Single molecule fret is a whole nother realm in the fret landscape. The capabilities that this application brings is that it can follow single event, like stochastic events. Like in this particular example, in this hairpin, if you modify the hairpin with two dyes, two fluorophores, upon the docking and the undocking, the two fluorophores are brought to each other, close to each other for fret to occur. So this increase that you see here in fret, the increases that you see here in fret are actually reports on single events as fret, occurs on and on. So this example here of single molecule fret is another extension of fret that can give you very, very interesting insight into single molecular dynamics. So you can take the information from single molecular fret and extract kinetics. For example, here, if you follow these events and you report occurrences as function of dwell time, you can extract these rate constants that you see here. So you see that single molecule fret will give you access to intimate kinetics that you cannot have otherwise with other systems. Finally, and to recap, and I know I went a little bit over time, fret is a process that is the result of non-radiative energy transfer between a donor and the acceptor, and this non-radiative energy transfer is extremely dependent on distance, and therefore it can be used to monitor interactions and dynamics at the molecular level. It is in effect a nanoscale yardstick or a nanoscale ruler. And with proper engineering, I hope I have convinced you that fluorescent proteins can be used to monitor temporal and spatial interactions within live cells. Finally, I would like to thank you for your attention, and I would like again to thank Dr. Midhat Ibrahim for the kind invitation. I'm looking forward to more interactions and more participation in this type of events. Thank you very much.
My name is Eugene Li. I come from Beijing Institute of Technology. It's a great honor to be invited by Professor Ibrahim to the 5th Hybrid International Conference on Molecular Modeling and Spectroscopy. I'm going to talk about my recent progress on the metal-based catalytic materials and their applications in electrochemical hydrogen conversion. Due to the serious pollution issues induced by the combustion-based automobiles, the whole world is now kind of in the process of transition into electrifying their vehicles. And China has made a great effort in doing this. And not only in China, the whole world is kind of have their time frame for the carbon neutrality. And previously, we used methane and then moved slowly to methanol. And finally, we believe we're going to step into a hydrogen economy. The conversion of hydrogen into electricity in automobile and portable applications has been considered as a dominating way to use hydrogen. More recently, the green hydrogen produced mainly from renewable energy such as solar energy and wind as energy storage medium is making its way into broader applications such as boat stationary power. Proton exchange membrane fuel cells is considered as a dominating electrochemical device to convert hydrogen into electricity. It has a symmetric structure separated by the proton exchange membrane. Catalyst is the most essential material in fuel cell by accelerating the reaction, especially at the cathode, known as the oxygen reduction reaction, which suffers from the intrinsic low kinetics. Therefore, developing high-performance ORR catalyst is essential. Currently, platinum group metal catalyst is the only usable catalyst in commercial fuel cells. For ORR catalyst, activity and stability are two essential factors to evaluate the performances. To design highly active ORR catalyst, we carried out investigation from facet engineering, defect engineering, near-surface engineering. And stability-wise, we carried out work on nanostructure engineering, alloy design, and grain boundary engineering. However, for the activity and stability, it is almost impossible to have it both ways. For highly active catalysts, they usually degrade fast, while for the highly stable catalyst, it usually has low activity. For alloy-based ORR catalyst, the transition metals will usually dissolve to form a stabilized PGM-rich surface structure in operation condition. Therefore, we develop a top-down selective etching method to establish a PGM-rich stable surface on palladium copper nickel ternary alloy. We also found that the underlying transition metals in the near-surface layer should have an appropriate ratio to regulate the surface structure at the surface, which corresponds well with the experimental discovery. In periodic table, there are several columns of metals that haven't received much attention in catalytic research or by the catalytic community. These elements feature with high melting point, high hardness, and most importantly, corrosion-resistant nature. For instance, the tungsten incorporated in the nickel's phosphor film has greatly inhibited the corrosion current, which has been used a lot in surface coating technology. We are one of the first few groups to use high melting point metals in ORR catalyst design. As an example, we developed a tungsten surface doping method as a bottom-up stabilization method to enhance the PGM ternary alloy stability. Based on the selective etching method as previously mentioned, we further dope tungsten on the surface, which can stabilize the weak points at the surface and deactivate the dissolution channel at the surface. We discovered that the surface doping of tungsten is accomplished through a surface reconstruction mechanism. The tungsten surface dopants are effective in improving the stability on the platinum copper nickel alloy. We further think beyond the surface doping and consider if tungsten alloyed in the bulk can further enhance the stability. The platinum copper alloy is employed as the model catalyst. By developing a nucleation trapping wet chemical method, tungsten can be successfully incorporated into the near surface. It is interesting to find that the tungsten can greatly improve the stability of surface area as well as the activity. We discovered that the platinum tungsten and copper tungsten both show higher bonding energy than the platinum copper, enabled by the electronic donating of tungsten, leading to the ionization of metallic bond and hence the improved stability. Using the same or similar design philosophy, we used palladium to dope the ultra-thin platinum nanosheet, and it is interesting that we find that palladium tend to dope at the grain boundary of platinum nanosheet. It is also discovered that the catalytic stability of the nanosheet is greatly enhanced. Next, I'm going to talk about our recent work on defect engineering of PGM catalyst. It is well known that as a potential hydrogen storage material, palladium suffers a big challenge of structural instability after repeated absorption and release of hydrogen. Basically, the structure defects will form after cycles of hydrogen storage. For hydrogen storage applications, this instability hampers its way to practical application. But for the catalytic applications, the defects could be favored because they may introduce unique performance towards the OR. Inspired by this idea, we developed an electrochemical technique to regulate the hydrogen absorption and release, as long as the hydrogen absorption and release behavior can be resolved in the cyclic voltammetry. Using palladium molybdenum as a model catalyst, the hydrogen absorption can be achieved by holding the potential at around 0.1 and released at slightly elevated potential at close to 0.08. Besides, the accelerated hydrogen absorption and release process can be repeated by applying cyclic step potentials. It can be found that the OR activity can be improved through the electrochemical hydrogen treatment. The mass activity can be boosted up to over 3.0 amps per milligrams of palladium. We first throw doubts to the real role of hydrogen absorption treatment and think that the activity enhancement could be induced by the de-alloying effect. However, the corrosion potential of the palladium molybdenum is found to be close to 0.4 volts, much higher than the treatment potentials. It is also possible that palladium hydride material could lead to the activity enhancement. So we synthesized palladium molybdenum hydride and found that its activity actually decreasing instead of increasing. Therefore, the enhancement can be largely induced by the hydrogen absorption and release treatment. It is well known that the activity is largely related to the geometric factor. Therefore, a synchrotron-based X-ray diffraction is carried out to investigate the strain distribution in the nanocrystals. It is found that the diffraction peak after the hydrogen treatment is significantly broadened, which means that there is microstrain in the nanocrystal. Using the Williamson-Hall method, it shows that a 2.4% localized surface distortion is generated that leads to the presence of surface defects. The geometric phase analysis on TEM results shows that a strain is present at the surface of the nanocrystals, strongly indicating the presence of surface defects at the surface. To confirm the existence of defect structure, positron annihilation lifetime spectrum is used. It is currently the most effective technique to investigate the defect in a statistical way by using the synchrotron-based facility. Tau 1, Tau 2, Tau 3 are three parameters to evaluate the nature of defects, which corresponds to the perfect lattice, atomic scale defect, and macro defect, respectively. It can be found that with the elongated hydrogen absorption treatment, the tau2 value is increasing while the tau1 is remaining almost the same. It means that atomic scale defect could be the dominating defect in the catalyst after the hydrogen absorption release treatment. The as-treated catalysts are employed in the zinc-air battery as a cathode or positive catalysts. It shows the device shows a good power density performance with fairly good stability over tens of hours, and this is an example of defect engineering. For the PGM catalyst, we developed a general electrochemical technique to introduce the surface defects. Apart from the commonly used proton exchange memory fuel cell that operates by air, the hydrogen-oxygen fuel cell is also important in special applications such as the air-independent propulsion submarine system or space unmanned missions, where oxygen is easier to obtain than air. For the hydrogen-oxygen fuel cell, to decrease the PGM loading amount, serious mass transfer issues may exist due to the higher transfer resistance of oxygen and low proton conductivity. Inspired by the design of hybrid catalyst with PGM nanocrystal and a single atomic catalyst that can enable a cascade catalytic mechanism, we intended to develop a hybrid catalyst to enhance mass transfer. Tungsten oxide has been utilized as proton storage medium due to its cycling capability of protonation and deprotonation. Besides, tungsten oxides have been considered as an active hydrogen spillover surface when being contacted to PGM nanoparticles. They have been demonstrated at anode or cathode catalyst in fuel cells. When using the tungsten oxide material in PGM-based alloy catalysts, we are surprised to see that the diffusion limiting current density is increased, which is a repeatable experimental finding. Does it increase the oxygen solubility or doing something else? We designed a hybrid catalyst with PGM and a tungsten single atomic catalyst to investigate the origin. By using a two-step mild synthetic method, a catalyst with a platinum nickel tungsten ternary alloy loaded on carbon support is synthesized. Interestingly, the carbon surface is covered with single atomic tungsten site. More interestingly, we did not find any nitrogen on the surface, which is the common ligand atom to stabilize the single atomic metal center. Instead, oxygen is discovered surrounding the tungsten. So we believe that the single atomic site on the surface is a tungsten oxide species. Besides, the platinum nickel tungsten nanocrystal displays a core shell structure with tungsten mainly on the shell layer. Compared with the platinum nickel catalyst from the first synthetic step, it can be seen that the platinum nickel are receiving electrons from the surface tungsten atoms coming from the second synthetic step. Besides, the tungsten displays a valence of 6, indicating that it exists as oxidized state. In hydrogen-oxygen fuel cell, with the above catalyst used as a cathode material, its performance increased by over 64%, with increased and enhanced stability as well. It should be noted that the PGM loading this work is only 0.05 milligrams of platinum per square centimeters, compared with the commonly used loading of 0.3 to 0.4 milligrams per square centimeter. The peak power density is over 2 watts per square centimeter, which is among the highest performance values. It is interesting that the diffusion limiting current is also higher than the normal values. After the stability test, the diffusion limiting current goes back to normal. This indicates that the enhanced diffusion limiting current may associate mainly with the tungsten oxide site that tends to be partially dissolved after the stability test. Besides, it could also be possible that the higher diffusion limiting current may have something to do with the more efficient proton transport facilitated by the tungsten oxide site. After the stability test, it shows that unlike the obviously larger platinum nanoparticles induced due to the like Oswald ripening or the coalition, the platinum nickel nanocrystals show similar size to those prior to the stability test. It corroborates the high stability of the as-designed catalyst. Besides, we also measured the oxygen transport resistance when using this catalyst in the device, and we discovered that oxygen transport of the hybrid catalyst is obviously lower compared with the platinum nickel catalyst that does not have the tungsten oxide single atomic site, meaning that oxygen is transporting much faster in the catalyst. We investigate the mechanism mainly from two aspects. For the improved ORR kinetics, we discovered from the density functional theory that oxygen actually binds much faster and easier to the tungsten oxide single atomic site, which could quickly initiate the oxygen molecule activation into OOH species that can quickly transport to the PGM surface at the vicinity, as well as the following steps. While as for the accelerated proton transfer, we discovered that the transition state between the single atomic tungsten oxide site is only 1.58 eV, which is much lower than that on the tungsten oxide bulk tungsten oxide surface, over 2 eV, leading to the boosted proton transfer in the catalyst. This is pretty much the main contents of my presentation on the recent work by using the high melting point metal in the design of highly stable and high performance PGM catalyst. I would acknowledge my collaborators, especially Professor Ke Xiaoxing from Beijing University of Technology, our Brotherhood University in Beijing. I also thank the PhD students who contribute to the reported work, as well as the founding agents. Next, as an editorial member of a new journal, SmartMath, I want to encourage your submission of your work to this journal. This journal is published by Wheelie, and it has very strong editorial board and editorial team. And its first impact factor this year is 20.4, a very highly targeted journal. This is another journal called Tungsten. As you all see, I did some work on using tungsten in functional materials. This journal is actually expecting more work on non-ferrous metal, not only on tungsten. Your submission is highly welcome. And also, this is a journal that I'm recently organizing a special issue. This journal is called Progress in Natural Science, and Material International. It is it is a bi-monthly journal published by Chinese Material Research Society, and I'm organizing a special issue named as Advanced Material Technologies for Electrochemical Energy Storage and Conversion. And you are all welcome to submit your recent work. And the deadline for this special issue is December 21st, 2023. I guess this is all for my talk, and thanks for listening. There may not be a chance for questions or communication this time. I listed my email. You can send me email for collaborating or communication or application for those students who are interested in pursuing your PhD or graduate studies in China, and your application to PhD in Beijing Institute of Technology is highly welcome. And thanks a lot.
Good morning to everybody. Thank you for the organizers to invited me again in this very fantastic conference regarding the fifth Hybrid International Conference on Molecular Modeling and Spectroscopy. I would like to thank in particular the Professor Meda, the Professor Anan, that I know them since several years, and thank you for listening me. The title of my presentation is Pharmaceuticals as Emerging Contaminants of River Ecosystems, Old and New Challenges. As somebody already known, my institute is an Italian institute of the Water Research Institute of the National Research Council in Italy that has 5 branches. The headquarters is in Rome, but other branches is 2 in north of Italy and 2 in south of Italy. I would like to start for the One Health concept. So a single health for people, animals, and environment. This is a very important concept in which all the health are strictly connected. So the health of the people is strictly connected to the health of the environment and the animals. In fact, the evolution and substance of our planet is based on a symbiotic relationship among humans, animals, and environment. The 3 blocks are interconnected. Humans are urgently to take into consideration the environment health and the animal health. In fact, we are increasingly vulnerable to contemporary global health challenges. But this time I would like also to focus on the antibiotic presence in the environment and antimicrobial resistance. It is not only a human health question, but also an environmental problem. If a threat occurs in the environment or in animals, this can have a consequence for humans. On this basis, on the One Health basis, it is easy to understand that all factors improving the presence of resistance determinants in the environment or in animal pathogens could affect human health. This is due to the flow of bacteria and genes among animals, people and the environment. Antibiotics are naturally present in the environment because they are produced by bacteria and to fight against each other, and they are also produced by some fungus, for example. But the antibiotics that are produced by humans highly increase the concentration of these molecules in the environment. So antibiotics can enter the ecosystem in various ways, from industrial wastewater, hospital wastewater, livestock, agriculture, or domestic effluents. Although some of them can be degraded or can remove it from the environment, not all of them are susceptible to this process of the degradation, and their constant introduction to the environment, that is so-called pseudo-persistence, has generated awareness in the scientific community. Antibiotics and related resistant genes have long been recognized as emerging pollutants. In fact, their regulation and entry into the ecosystem have not yet been controlled and regulated. Recently, in Europe, we have the Water Framework Directive, that is a very large directive that comprises the regulation of the contamination of water. At this moment, we have only watch list that I listed in the following presentation. About one of 5 resistant infections in humans are derived from food and animals, and this is a very important point. Antibiotics are, as I told you, are only partially metabolized in the treated organism, that is humans or animals, and through excretion they reach wastewater treatment plants or soil. Most of wastewater treatment plants are not able to remove them efficiently, and through the effluents they reach surface water. The use of sewage sludge as biosolids and manure as crop fertilizers and irrigation with the reclaimed water from wastewater treatment plants will entrain human and veterinary antibiotics into agricultural soil. A transfer of antibiotics and also antibiotics-resistant genes from wastewater or animal manure through soil to edible crops is also possible. As I told you, the European Water Framework Directive include a watch list. The first watch list was released in 2015, and the watch list included a list of contaminants that are at the moment checked in the water, but are not at this moment included in the list of contaminants that all the countries, European countries, have to analyze. But it is a list of contaminants that probably will enter in the list of contaminants, regulated contaminants. Since 2015, antibiotics were included in this list to be 3 years later, some antibiotics, macrolide antibiotics, together with other antibiotics, were included in the watch. The third watch list also included 4 antibiotics And the last watch list that we had, that was released last year, also include for antibiotics. So these antibiotics were checked in different water, surface water or groundwater in some countries, and before the analysis of these contaminants, they probably enter in the list of the contaminants that have to be analyzed from all the European countries. Also, I told you one in agroecosystem antibiotic contamination can be found also in soil. At our institute, we have performed several experiments in laboratory that included some fluoroquinolone and sulfonamide antibiotics and their effects on natural microbial community of a river, in particular Tiber River. Why we analyze this effect? Because the natural microbial community can degrade the contaminants and also the antibiotics. But if the concentration of these antibiotics is high, they can kill some important microbial groups that are involved in the ecosystem services. The increase in bacteria-resistant population also is an important factor that we analyzed. And also the presence of the antibiotics promoted the degradative populations. Here you can see a list also of some papers that we have already published. But what about the resistant genes? And the presence of resistant determinants in pathogens on environmental bacteria can be due to different factors: DNA self-mutations or genetic material acquisition from other microorganisms, including the environmental one and the clinical strains. And this acquisition, performed by horizontal gene transfer, has indeed been found between environmental and pathogenic bacteria, mediated by mobile genetic elements such as transposomes, plasmids, and other Antibiotic-resistant genes have existed since a long time, and antibiotic-producing species, for example bacteria, are common in natural microbial communities. So antibiotic-resistant genes naturally occur in environmental microbes. But native resistance does not pose a threat to human and animal health. If antibiotic-resistant genes are present in clinical important pathogens, the antibiotics are no longer able to kill or inhibit the growth of these bacteria, which translates into a problem of huge proportion. Soil, river, and their sediments are recently being recognized to be a potential source of transmission of antibiotics. Although the antibiotic-resistant concentration found in the environments do not pose an acute risk for human health, the long-term effects of antibiotics or antibiotic-resistant genes are not clear. And which are the environmental side effects of the presence of antibiotics and resistant genes in the environment? Residual concentrations can promote antibiotic-resistant gene spread, and the residual concentration can modify the natural microbial community. So the natural microbial community of water and soil ecosystem have a key role in different cycles of mechanism or process and for the maintenance of ecosystem quality, comprising fundamental ecological processes, which is the concentration that promote the antibiotic resistance and which is the basic level of resistant genes in the environment that does not pose a risk. So these are the two main questions that we have to respond. The environmental concentration of antibiotics are in the range of the so-called minimum selective concentration, which can stimulate the evolution and dissemination of antibiotic resistance. And this concentration can act in three different ways: selecting resistance, making genetic and phenotypic inconsistency, and signaling molecules. Which are the old challenges, but at this moment are not already resolved. Wastewater treatment plants, from domestic ones, from hospital or from pharmaceutical factories, are recognized a crucial point of pharmaceutical contamination, and urban wastewater treatment plants are an important reservoir of antibiotic resistance. In some cases, tertiary treatment have been found to be more effective for pharmaceutical abatement, including antibiotics and antibiotics-resistant genes. So the tertiary treatment of the wastewater treatment plant can be very useful for partially decreasing this. In this slide, you can see the pharmaceutical pollution all over the world in different rivers, and the concentration that were found. This is a publication of last year, were lower than the concentration that can cause ecological effects. The exception was for sulfamethoxazole and other pharmaceuticals, but in particular, I would like to highlight on sulfamethoxazole, that is an antibiotics. A concentration of 9 of the 13 detected antimicrobials exceeded the safe concentration recently proposed for at least one sampling site, with ciprofloxacin, that is another antibiotic, exceeding the safe limit at 65 sites. But we have also some good news. In fact, the European Union withdrew the antibiotics as growth promoters since several years. In America and in China, banned the use of antibiotics as growth promoters as veterinarian growth promoters. So this is a very important point, because in the past antibiotics were used very largely. In India, 3 years ago, announced stringent standards of concentration of antibiotics found in the waste discarded by pharmaceutical factories into rivers. And the new regulation of European Union on veterinary medicinal products entered in force last year provide all forms of routine antibiotic use in farming, including preventive group treatments. So European Medicine Agency reserve antimicrobials for humans to combat rising superbugs. Other good news. Microbial biodiversity and well structured can limit long-term invasion of antibiotic resistance, but loss of soil microbial diversity exacerbates the spread of antibiotic resistance. Only high amounts of antibiotics was found to promote resistance in a long-term study performed in Canada. So not in all the conditions, we have the promotion of the resistance. In our institute, we tested the effects of antibiotic sulfamethoxazole, ciprofloxacin, and enrofloxacin on soil microbial communities for assess the degradation in a cattle digestate amended soil and to assess the antibiotic-resistant genes spread among soil microbial populations. We tested the environmental fate of resistant genes antibiotics in digestate in these papers, and we found that an average of resistant genes was found in this experiment, but the antibiotic and RGs concentration were significantly lower in the digestate if compared with the manure. So a good news is please use the digestate from digester plants instead of manure. If you use this fertilizer in agriculture. Antibiotics and resistant genes in Lactuca sativa leaves following manure and digestate application to soil and effects on microbial community. We performed these experiments and we found that also in this case digestate showed to be an organic amendment more suitable than the manure, decreasing antibiotics and antibiotic-resistant genes. And further composting aerobic process could further improve the digestate quality, and these studies are actually in our institute. New challenges: how to measure the antibiotic resistance, because chemical analysis of the antibiotics in soil and water are not enough. So several research have been found antibiotic resistance independently from the antibiotic presence. So it is important to analyze not only the antibiotics but also the resistant genes, or, for example, some mobile elements that can be transmitted the resistance from the environmental bacteria to important other bacteria. Because no single chemical or microbiological parameter can be used to infer the antibiotic resistant load, its specific monitoring should be part of the parameters used to assess wastewater quality. As I told you, in fact, the wastewater is a crucial point to the spread of the antibiotic-resistant genes. Other new challenges. Other contaminants can improve pharmaceutical movement and resistant gene spread. In this case are the microplastic. In water and soil, they can absorb different contaminants, including antibiotic-resistant genes. So it is important also to consider not only these contaminants, so antibiotics or pharmaceutical in general, antibiotic-resistant genes, but also the presence of microplastics. The disinfection of drinking water also, for example, the chlorination, was found not effective for antibiotic-resistant genes abatement in drinking water. So this method, unfortunately, is not able to abate resistant genes. Latent genes and antibiotic-resistant genes can be a problem Latent antibiotic-resistant genes are abundant in all environments. The core resistome in humans, animals, and wastewater consists of a substantial number of latent antibiotic-resistant genes. Many latent antibiotic genes are associated with mobile genetic elements shared between environments and present in human pathogens. And how the coronavirus disease pandemic and climate change contribute to pharmaceutical contamination? The climate change can enhance soil or water ecosystem contamination by antibiotic-resistant genes. In fact, the climate change, so the increase of the temperature, can be considered a threat multiplier for the spread of infection, disease and antibiotic resistance. The increase in temperature due to climate change is a key factor for bacterial survival in the presence of antibiotics. Therefore, it is essential to understand the relations between temperature changes and selection of antibiotic resistance. Transient physiological response to temperature may alter cellular behavior and lead to antibiotic tolerance and persistence. In fact, there are similarities between antibiotic physiological effects and adaptation to thermal stress in response to increasing global temperatures. Also, genetic responses such as the appearance and conservation of antibiotic-resistant mutation may be in part due to thermal stress, and large-scale responses, local and global changes in temperature are associated to the spread of microbes that may cause human illness. Some antibiotics and resistant genes can be found concentrated in sediment or rivers. Here, they persist for long periods. Sediments could consequently act as antibiotic-resistant genes reservoir. In a world of changing water availability that can worsen due to climate change, it has to consider antibiotic-resistant genes in sediments that may be resuspended during floods, making antibiotic-resistant genes more available and enhancing their resistance by availability to disease causes microbes. This fact was already found in general for other contaminants, but not for resistant genes, and future studies should be conducted to further investigate this effect during floods. An increase in precipitation intensity has the consequence of the runoff of contaminants from soil to water, or on the increase in river flow on the amount of organic substances. On the contrary, the prolonged absence of precipitation can concentrate contaminants, in particular in intermittent water bodies, and have a stress effect on microbial communities. In both phenomena that are very common now in Italy, but I think also in your countries, in Egypt or in other countries, the nutrient cycle and the biodegradation of contaminants, including antibiotics, can be altered. But also the antibiotic resistance can be further developed by the COVID-19. The first aspect is the overuse of antibiotics. The presence of some chemicals, such as metals or biocides, could enhance the development of resistant genes and the increased use and overuse of antibiotics and biocides could enhance by antibiotic resistance. This results in an increase of antibiotic residual concentration in rivers, receiving effluents from human and hospitals, wastewater treatment plants. several individuals with a mild or moderate disease are treated for prophylaxis or treated purposes with antibiotics, and several diagnoses for COVID are associated with bacterial infection, are the majority of patients are treated with antibiotics, even when non clinically indicated. So a very high concentration of an antibiotic in river is a consequence of this different treatment. And a second aspect is the overuse of biocide. The wide use of biocides for environmental and personal disinfection, including in non-health care settings, could enhance the antibiotic resistance, and low exposure to biocides can select for antibiotic-resistant genes and enhance the risk of cross-resistance to antibiotics. Potential environmental impacts of elevated biocide use include several effects on wastewater treatment unit operations, antibiotic-resistant proliferation, and high formation of disinfection byproducts. Finally, the problem of exposure to antibiotic resistance has to be taken very urgently into consideration, especially in situations with low access to clean water and in countries at war in which there are aggregate people without access to hygiene, sanitization, and safe drinking water. Some concluding remarks. The use of pharmaceuticals has been resulting in drug presence in all the environments. The use of antibiotics is an important factor in the spreading of prevalence of antibiotic-resistant genes as well as of future emerging resistant pathogens. Combating antibiotic resistance in the view of One Health and global health perspectives will be successful control of antibiotic resistant genes in the environment and human and veterinarian level. All the action at local, national, or international level preventing the input into the environment of antibiotics or other cofactors such as other contaminants, stimulating the presence of the antibiotic-resistant genes is of fundamental importance. And wastewater treatment strategies for diminishing antibiotic and antibiotic-resistant gene spread need to be urgently put in place. Monitoring of antibiotics and their resistant genes at environmental level is of great importance. It is the first step towards a real contribution to the protection of ecosystem, animal and human health. And the minimal selective concentration for antibiotics could be a first step towards identifying environmental important concentrations. Isolation or sequencing of microorganisms would be necessary to identify specific markers, mobile elements, and genetic backgrounds for more accurately tracing genes and assessing the risk posed by these antibiotic-resistant genes. There is a need for more research on the environmental fate, toxicity, and antimicrobial properties of antibiotic metabolites that are not at the moment tested, including those from wastewater treatment plants, which in some cases may be more persistent or with a greater capability of producing antibiotic-resistant genes. These are some of recent papers regarding the antibiotic presence in the environment and different experiments that we perform in our institute. And thank you for your attention.
Assalamu alaikum and greetings from the Ohio State University. I thank you for the opportunity to come back to this, my favorite conference. And today I'll be talking about what we have found in solving toward the solar opacity, which is related to the solar abundances. It's a long-standing problem. If you okay, this is the work accumulated for a number of years to work on it, and finally we have submitted 4 papers at the same time. And whatever I'll be presenting is the contents of the second paper, and largely to study the atomic process of photoionization. And along with me, there are 2 other people who contributed a lot. One is Lianxu Zhao and Werner Eisner. So as I mentioned, these 4 papers in work for quite a number of years, and it discusses mainly the physics of solar opacity. And the modeling will be the next part. But why solar opacity is so important? Because opacity is a fundamental quantity for plasmas. So it gives a measure of radiation absorption. And the main thing is that it is related to the abundances of elements in the sun. And we focus on the opacity at a particular region inside the sun, which is the boundary between the radiative and the convection zones inside the sun. So why the boundary? The reason is that this boundary is known very accurately from helioseismology measurement. So if the opacity is accurate, then it can be used to predict where this boundary should be following the optical depth conclusion. So, and why we are studying iron in this particular region? The reason is that iron is abundant in that at the boundary of this convection and radiative zone inside the sun. And not only that, it is the main source of radiation absorption. Opacity is actually means radiation absorption. So, ions have large number of energy levels. So if it can absorb many photons and causing a huge amount of radiation absorption and giving the opacity a measure of the plasma. And we have been using R matrix method for this iron study. Question is again, why R matrix method? Because it is the most powerful method to study atomic processes. This and their characteristics, there's a way when you solve the equation, this method solves the space in our equations by dividing the whole space into 2 parts, inner region and outer region, where the atomic species is inside the region and the outside is relatively free. And somehow it connects and it can cover a lot of energy levels and other computations. But the computation is very, very extensive in this method. And what are the new physics that can impact opacity? That's the reason we studied and we have found and written up the paper, the new physics which are very, very much relevant to the opacity of the solar plasma. So this slide shows the definition for opacity. So when radiation passes through the plasma, they get absorbed by the elements in the plasma and then emitted out. So during this process of absorption and emission, the radiation loses energy. So the intensity becomes weaker. So if we take the ratio of the intensity, incident and the emitted intensity, we see it is it depends on the exponentially on a factor called tau, and tau depends on kappa, which is the opacity, is the fundamental quantity, and rho is the density of the plasma, and x is the depth or the region of traveling. Because this opacity is so much prevalent inside the sun, is the reason that the radiation created at the core of the sun takes over millions of years to travel up to the surface of the sun to escape outside, because they get absorbed and emitted by the elements. Okay. So it just shows an example how opacity calculation started, why it is so important, and how two projects are now initiated just because of that. One is called the opacity project, the other one is called the iron project. The opacity, because in the past, when the pulsation of Cepheid variables were measured and being predicted, they did not quite agree. This is the measured pulsation. Well, it is the ratio of the periods pulsation, and if the existing opacity was used, that is what they're getting. And now opacity is also related to these pulsation periods. So they knew there's a huge missing part in those existing and opacities, and that's what the opacity project started, and it carried out the first most elaborate calculation of the atomic processes for accurate opacity. And it did solve many problems. And not only that, it developed theories, and many people are involved. It was led by Professor Michael Seaton of University College London, and it ended up 2 books, and this is the volume 1 with a lot of papers, and people involved, and over 50 people were involved in this computation. And we found a new physics, and there are a lot of publication, and we created 4 databases like TopBase, PanticBase, OP-Server, and NORAD Atomic Data, which is at Ohio State University. And the problem was solved. You can see that a pulsation, this period is now agreeing with the predicted line. This is the earlier opacity prediction, and this is the newer one. But with all this solution, not all the astronomical problems are solved. The main question was with the sun. It just turned out that we have a big problem with the sun, and it was expressed in a publication by Buckle in 2004, I think. So what it was, the finding was that the opacity they needed to be increased for the sun, because it was not giving the right numbers for the abundances. So if the sun has this core where the nuclear fusion is happening and giving out high energy photons, they are traveling in the radiative zone and very dense plasma. After that, this boiling plasma region, which we call convection zone, and this is the boundary. And this boundary is known very accurately from heliosismology, and from the center and up to that boundary is 0.713 relative to the center, and plus minus 0.001. So it's very accurate. And however, if we use all the atomic data to calculate the opacity and then predict the boundary, it comes out to be at 0.726. Although these two numbers are not much different from each other, but given the size of the sun, small difference translates into 5,600 miles. That means this boundary has expanded by 6,500 miles around here. So prediction of the sun is that it is much bigger than actually it is. So this is the problems to get this proper values, we have to increase the opacity of the sun. At the same time, it turned out that the abundances we are concerned should be lower than what we think. The 3D models, which study the sun, inside the sun, and it shows that the elements like carbon, nitrogen, oxygen, they should be lowered by 40 to 50% than what we know today. So all these discrepancies indicate that there's a big missing part of physics in all these calculations and predictions. So that is what we are focusing on to solve this discrepancy. So opacity depends on two main processes. One is a radiation absorption by excitation, which we call photoexcitation. And because energy levels are quantized, so that's why we can see either fixed photons at fixed energies, which can be represented by these lines now in the spectrum. And also photon can be absorbed by this atom or element for ionization when the electron is ejected out. So because this is a continuous process, electron goes to the continuum, the cross-section for ionization is a smooth curve. But when it hits the threshold, and various ionization thresholds, so we usually have a peak like that. However, in between those, there are some quantum states. So I'm explaining in this energy diagram. This is the energy diagram for the element, and this is the continuum. This is the ionization threshold. Beyond it is all positive, but it also has quantum states. These quantum states are called Rydberg autoionizing states. They form quasi-bound states. So if the photon energy matches with one of these energies, what will happen that there'll be doubly excited states, and the electron will eject it out ultimately. But that intermediate state will introduce a resonance, say, on the cross-section. And we'll see lines. These lines, which we call cross-section, can be seen in the absorption spectrum. If the resonances are strong and they are relatively isolated. Otherwise, they often get dissolved by the density and the temperature of the plasmas. I'll show example. In this example, so this is the photoionization cross-section of iron-17. This is without any external effect. But if we increase the plasma around it with density, increase the density higher and higher, these lines slowly get dissolved, and they keep on dissolving at some energy, some high density. All these resonances will dissolve into flattened line like this. It doesn't mean the cross-sections have gone away. The area underneath here is exactly the same area underneath here, only they have flattened out. So we know that that's the effect, plasma effect on the resonances of cross-sections. So the solar abundance problem was also studied at Sandia National Lab at the Z-pinch experimental setup. What they did, they created iron plasma at the same temperature and density as in the sun in the laboratory. So they created a small, tiny sun inside the laboratory and measured the opacity by passing radiation through it. And that is what they measured, the black curve. And the cyan curve is the predicted opacity using the opacity project data. And we can see that the peaks have some sort of agreement, but the background is consistently lower, and there is also a big window here and there. So that means the data that we have currently are not accurate enough to solve or to give the accurate opacity inside the sun. So we wanted to find what to create this cause. We did repeated the calculations for photoexcitations of the ions, and also we needed to also close this gap from, and these gaps comes from photoionization, as in like here, the cross section and are flattened out, so we don't see the peak. So we studied the photoionization cross sections and our report our findings. But in that particular energy or the plasma state inside the sun, at the convection and radiative zone, the temperature is 190 electron volt, and the density is 10 times 10 to the 23 particles or electrons per centimeter cube. At that temperature density, the surviving ions of iron are the three ions: iron 17, iron 18, and iron 19. So these are the three ions which are dominant in the plasma, and that's what we studied, photoionization of these three ions. Okay, I'll show what we did. When we calculate photoionization, we know that the temperature will excite the ion to various excited states, and that means the outer electron will be at various excited states. But the core ion inside can also be excited state. And this, when the core ions are excited states, that's when the resonances form. So what we did, we took this core ion into many excited states. In general, it is assumed that this excited state will not form any bound state. It is not an assumption, that is what is known. Actually, we see that bound states don't form with many highly core ion excited states. But we wanted to see what will happen if the core ion is at various excited states. So this is an energy levels of the core ion. So it's a ground level, and that's the excited 9.4, and then the next one is 9.8 Rydberg. But then this is for n equals 3 states. For n equals, no, sorry, 2 states. For n equals 3 states, there's a big jump. Instead of from 9.8 Rydberg to 56.7 or 8 Rydberg. So the big jump, and there are a lot of states at N3 complex, N3 levels, and we have included 110 levels from N3's complex. And not only that, we went beyond that when what happens if we include more states for n equals to 4 levels. So we included 105 levels, and then these are the levels that we included to study photoionization of iron 17. We did similar kind of calculations for iron 18 and iron 19. And our purpose is to calculate the cross section. So the transition matrix element is given here, the initial state wave function and final wave function. And this is the interaction, the photon interaction. So if we take the mod squared, we get the line strength. Once you get the line strength, we can calculate other quantities like oscillatory strength, radiative decay rates, but we need the photoionization cross section. So with the line strength, and this is the EIJ, that is the transition energy, and GI is the statistical weight factor. But how we can incorporate all those excitation of the core ion and in our calculation? There's only one way we can include all those excitation, 218 excitations of the core ion, is representing the ion, which is iron 17, in close coupling approximation. This is the expression for close coupling approximation. This term is basically here, but just separate out to show orthogonality. So main difference of this wave function compared to other wave function is that we write the total wave function as a function of the core ion wave function, which is chi I, multiplied by the outer interacting electron wave function. In this way, we have two wave function interacting, so electron can be free. In that case, the energy will be, the end up an eigenvalue will be positive, or electron can be negative, the energy will, that means it will be a bound state. And this, the core ion, it could be at various states. So for the wave function of iron 17, for example, we had 218 levels. So here, okay, then we solve the Schrödinger equation. When you put the wave function, we end up with set of coupled equation and gives a Hamiltonian matrix, and that matrix is solved by this method called the R matrix method, which now has a way to solve a complicated system, and that's what we have used to study these ions. Okay, and this figure shows the various computational stages that the R matrix competition needs. And now, like, we have stage 1, stage 2, recap D, various stages. And these are the three ways to calculate. We have chosen this side, which is called Bright-Pauli approximation, Bright-Pauli R matrix method, to calculate the dipole matrix, Hamiltonian matrix. Ultimately, the products are like energy levels, oscillatory strengths, photoionization cross-section, reconversion cross-sections, and collision strengths, which all go to the astrophysical modeling. Yeah. I'm just going to show the results. This plot shows the results of photoionization cross-sections of the ground state of the three ions, iron 17, iron 18, and iron 19. And you can see the features. In these features, we can see these lines, or the resonance lines. And in the low energy region, but the high energy region, they're all resonances are very weak or small, or they're almost merging to the background. All these resonances, they are created by the core ion excitation to the low-lying excited states. Okay, so that means if we include 218, not all of them are contributing to give any new features. Here, I can see only the very weak features. So for ground state, the high-lying excited state do not contribute. Okay, so this one shows another features of inclusion of relativistic effects in photoionization. And the photoionization cross. This is the photoionization cross section. Without the relativistic effects, we can see the resonances. But if we include the relativistic effects, the fine structure will split all the energies into various components, and each component will have a series of resonances. So that's why we can see, they are split into other resonances. Not only that, in addition to the relativistic effects creates those resonances which are actually observed, but cannot be created without inclusion of relativistic effects. So that is what we are predicting here, that we can see all these resonances experimentally. So this is one effect. The other effect is for equivalent electron state. These kind of states actually forms when there are more than one electron in the outer electron, and most of the atoms or ions have one or two equivalent electron states. These, even though they are very few, they are very important because they create very strong and high peak resonances. So these are showing, the resonances for iron 18, and you can see, the enhancement at various thresholds, but they all belong to the lowest N complex, N equals to 2 complex. But high-line complexes, they are not contributing because these resonances are quite weak. But the background decays very slowly. It means that this is a kind of cross-sections of equivalent electron states contribute significantly in their applications. Now the changes happen to all the excited states. Okay, most of the excited states are single electron excited states, and there are infinite number of them. And the thing that we consider the core ion excitation to high level excitations and have impact on these cross sections. This is the cross section of the first excited state with a single electron. Even though it is the first, now you can see the impact I'm going to explain. Now you can see starting from the ionization threshold, we can say very weak resonances. All these resonances belong to N equals to 2 complex. That means core ion excited to the N2 complex. But from here on, you can see the resonances are higher by 3 orders of magnitude, very strong ones. And not only that, the background is also here enhanced. All this means that the photons are being absorbed when these energies are reached. So that means more photons are absorbed. That means opacity is increased, which was not included. This part was missing and not studied before. The calculation gave only this simple line, and that's why the lower opacity happened, was obtained. But inclusion of this, a huge amount of radiation absorption, opacity is expected to rise. Okay, this figure shows not the low line, but the high line excited state photoionization for iron. This is for iron 19. We can see that again, similar features. We can see all these narrow resonances. These are like Rydberg series of resonances going up to N equals to 2, and we can see very high resonances. But in this resonance, we see broad features. These features are also something new that we found out under the opacity project. These broad features are actually resonances created, by, you see the airlines, there are something called Seaton resonances. These resonances can form a broad structure. That means huge amount of radiation can be absorbed in this region, and the whole thing has been enhanced by the Seaton resonances. Again, we can see that in all these resonances coming from N equals to 3 complex, but beyond that, after that, the resonances are decaying. That means no more strong resonances are found. They are just converging onto the background, telling that this contribution has been converged. So we have to make sure that the resonance contribution have been converged. The last figure I have on this plot is the highly excited states where we can see the features of Seaton resonances. But in those excited, there are 3 levels. Well, each one, this one is 3p, outer electron is 3p, it is 4f, and this is 7f. So that means, as we're going down, threshold energies are decreasing. That means we need lower energy photons to ionize them because they are highly excited states. But even though they are different excited states, but these broad features, which is caused by the Seaton resonances, they are not moving. They are remaining in the same energy positions. The reason is that the core ion only absorbing the photon to manifest this kind of broad features. It's nothing to do with the ionization. Ionization happens later on when the electron goes back to the ground state, and then outer electron goes out and photoionization happens and manifested in a broad region. So this is one aspect of this Seaton resonance. Second one is that as you go to a higher excited state, this broad region becomes more prominent. See here, this is the low excitation state, and this is the higher excitation state, and this the region is much higher compared to the background. Here, it is not the broader region is smaller compared to here. So these are the two features we want to say about the Seaton resonances. And I like to conclude that we have presented photoanswer cross sections of the three dominant ions of iron 17, 18, and 19. And the results are expected to reduce the discrepancy of the predicted opacity and the measured opacity, and also improve the abundances in the sun. And we got oscillatory strengths, but those have been reported. I haven't shown them here. And I've shown that the most extensive calculation up to date that we have carried out for was these three iron ions with a very large wave function, 218 levels for 17, 276 for iron 18, and 415 levels for iron 19. And we have seen that a huge amount of photon absorption, particularly for the highly excited state when N3 complex. But beyond that, there we see also notice that convergence. So we expect to all these new findings will solve or at least narrow down the existing discrepancies. Thank you. I can take the questions. So I'll stop the recording. Assalamu alaikum, and greetings from the Ohio State University, and welcome to a session of International Society of Muslim Women in Science. Let me share the file. Okay. The objectives of this society is to encourage Muslim women to science professions and retain them in STEM. And Islam encourages knowledge for both men and women. Today we'll inaugurate a chapter of ICMMS in Egypt, inshallah. STEM, which is science, technology, engineering, and mathematics, is part of life. It is needed for advances for humanity, for survivability. The knowledge gives us the power to solve problems we face, such as COVID-19 pandemic, climate, space explorations, health, agriculture, and gives us the control to shape our future for the best. So why females are important in STEM? They are the half of the population, half of the intellectual power to make advances. So women and girls are still underrepresented in STEM fields, and among all the races, Muslim fraction is the lowest, even though the total number we have is not very small. But we have many problems to solve for our advances. One big problem has been the climate. As you know, this greenhouse effect, the solar radiation will be entering the earth, atmosphere, heating up the atmosphere and the earth, and that amount of heat will also go back to space. But now, because of the capture of the radiation by the carbon dioxide in the atmosphere, the radiation stays there longer time before it is, it leaks out. So it means, it starts to warm up the atmosphere. And that has started since the industrial era started on Earth after 1850. So this plot shows that the temperature variation with time and going up to 2025, at this time, the temperature from the background, which is 14 degrees Celsius, has gone up by 1.2 degrees. So there, the world leaders are discussing how to cut it down, and so that we don't produce too much carbon dioxide. And for the present goal is to not go beyond 1.5, and by 2050. The water level has increased by 8.9 inches So we need all kinds of study, and we already feeling the disaster caused by this global warming. This picture shows the crack in this Arctic ice. That means the ice, the ice chunks, they will slowly break away and go into the ocean. So that means we'll be losing our drinking sweet water to the ocean. So this is a big problem. As we have to know how to regulate the carbon dioxide, still move on. Our research should continue in all sectors. We are lucky that this Professor or Dr. Shaheen and his wife, Dr. Ozan Toshi, they have been doing research with messenger RNA. And the COVID-19 exploded all over the world, and we lost almost 7 million people from 200 countries. The pandemic was devastating, and extremely unfortunate things happened, but it was their research that brought the first injection or the vaccine for COVID-19. So that's why we should continue our research in all sectors of STEM. The other part is that AI, the artificial intelligence, is becoming part of our life. It is doing a lot of data analysis because it can do that without being distracted, and it can sort out all the data and give us some kind of suggestion, like in medicine, disease, then they can give some suggestions. So in that respect, it's very helpful. Even Japan is using them as a friend to the elderly people. And on the right side, even AI, is helping Ohio State, this telescope with 500 cables. That is 500 telescopes. Each cable is capable of studying one particular object under the regulation of AI. So AI can do a lot of things, but at the same time, it can also make decisions without the ethic and human logic, which can be devastating for humankind. So we need to proceed very carefully. We need to know how to harness the power and capabilities of AI. Is very important, and also the stone and like asteroids and meteors, they are always passing by our Earth. This year, in other words, another big one came, and asteroid BU 2023. It didn't hit our Earth, but some of them do hit. But luckily, after that big explosion when the dinosaurs died, we didn't have that big one that caused a lot of damage and harm for the human beings. But when they come, we cannot even predict when it is coming, and if it is coming, how to stop it. So even though we have a watch and in the space by NASA satellite Scout, and we are doing some experiment, like a DIRT satellite, so that satellite can just crash into the asteroid so that it can deflect the path instead of hitting the Earth. This is still under a lot of investigation. We are not there yet. We need a lot of research in that. And the recent, and I would just say that this earthquake at Marrakesh is also devastating. Thousands of people died. It is in a short time, and there's just the village just depleted. It is so unfortunate and sad. Yeah, people are trying to pull out some more people. That we need to find a way to detect, when there is a vibration, tectonic vibration. We can have a satellite up there to monitor the vibration, or other kind of detectors on the Earth, so we can predict something. We are still behind in all respect. So all these things, we can go with the list, what we need to do to protect the human beings from disasters. So all we need to do, go to STEM fields. But the STEM fields is different from other fields because this discipline needs thinking, concentration, and perseverance. And sometimes this turns out to be a fear for others, but there should not be any fear, only that concentration and motivation are needed. for Muslim women, they are capable of, they're intellectually capable, and they're admirably inquisitive and deeply interested in exploration of science, of the universe, but they remain quietly behind the traditional family responsibilities and also the environments which don't encourage them. But it doesn't mean that the Muslim females have not contributed, and they have contributed to advancement. But each of us has to nurture and utilize our intellectual power. So we need support and time to detach ourselves from other responsibilities. Detachment is a necessity for any thoughtful work. The picture shows myself and when my son was 8 months old in a conference, and the picture was taken by my mother. This picture shows the United Arab Emirates University, where I gave the talk on ICMMS, and the girls in the audience, they were all beautiful, very lively, very fashionable, but they didn't want to have a picture with me after the talk. Only she had the hijab on, others didn't, so she took picture with me. But this is our tradition. What can I do? We should respect our own tradition. But general assumption is for science is that it is difficult, but nothing worth comes easy. So we have to make effort. So we can get 1 credit, and if we are successful, we'll get 2 credits. That's the thing I learned in school. And so it means we have to face challenge. We have to face the challenge and know the boundary of our talent and power. So I would like to say something notes around the charter of this ICMMS, that each of us, male and female, have biologically the same brain and the same capability to understand the amazing mysteries of nature, finding out the laws that govern them and implement them for our benefits and advancement. Even if we use our brain continuously, we would probably use only 10 to 15% of it. No religion restricts a person from learning, gaining knowledge. Knowledge is a necessity, and Islam encourages knowledge for both men and women. All living forms, the world we live in, the universe around us, have been created not to ignore them, but to know them for our purposes. The more we know the universe, the nature, the human functions, living beings, the more beauty we see, the more power and control we achieve to solve problems, the more we admire Allah and His creation. So this shows our contribution. Al-Azhar University is to be known as the oldest university, but there is another continuously running university in Morocco, which is called the University of Qurain. This university is a few 100 years older than Al-Azhar University, and it was built by a female named Fatima Al-Fihri. So that is a tradition. We should mention our contribution to others, advertise our contribution to others. It is important because other people also do that, other groups. So if we advertise it brings a respect for our group. So I also wanted to mention this particular picture. This is a madrasa built by Sultan Ulum Beg, but he was very much interested in astronomy, and on his own, he built the first observatory, modern observatory behind it, and he used to observe the sky and recorded new astronomical objects. So he's actually the founder of observatories. But we also face problems because there are people, we have less respect for our traditions and our values. And that should not stop us for going ahead. This shows, the BBC News published a few days ago, the French banning girls who are covering themselves up going into the school. So this is something not very positive and not respecting our religious values, our tradition. And but we cannot do something which can harm us. So, but we have to find other way to go move ahead or campaign to accept the way Muslims dress. Only not that, this is something I also face, but those things I don't keep in mind. I face things like people telling me that your prayers are like strange gestures. If I go to bathroom to Urdu and they look at me, because I'm splashing a little bit water outside when washing their feet, and they say also make comments about abaya or burqa, saying that it's a forced dress on women by men like a slave, and they make non-supportive statements about Muslims. And one of my students from Nigeria, she was telling me that she doesn't mix with Muslim female students because she thinks they don't value education. She is so much wrong, but that's I cannot do anything, and this is their values or their understanding. So what we need to do, we have to be united and do something. And I was inspired particularly by this professor at the center of this picture in front of Egyptian Museum in 2008. She was with me whenever we are going somewhere, and when she was leaving, I found out she was already a professor at the University of Khartoum in Sudan, and she was doing research without any contact outside, collaboration, no interaction with outside, on her own. She's devoted, but very little. When you do things isolated, advances are slow. So to make that, I thought, what can be done that can help researchers like her can contact outside and make the research a little bit faster. Not only that, and most of the Muslim females don't know how to cope with it, and modern, particularly Western, and women, female scientists, they have difficulty because of the cultural differences. So what I thought, making a society where everyone is free, the way they want to dress up, the way they want to talk or follow the tradition, and the environment is free. So because we are not different from other races, this is an example of ourselves. This is a function at Aligarh Muslim University in a female hostel, Sultan Jaha Hall, where I was asked to give handed out the prizes. And the first prize for dance was a girl who was standing next to me with the abaya on. I saw her dance, and she was competing, even probably exceeding Bollywood dances, and the fashion show going on were like compatible with French, fashion shows. But no man was allowed in the event. She was she was with the abaya because her parents were waiting outside to take her home. So she just waited for her prize and then go home with the abaya. I just looked at her and thought, such a wonderful thing. She's so lively inside, but outside she's so simple and modest. So abaya seems a modest dress to me and for many. So in 2010, I founded International Society of Muslim Women in Science. And during a conference in Egypt, actually, the objectives were that form a network to provide moral support, encouragement, retention in science and research, information on higher education, jobs, fellowships, secure grants. And our motto was, stay in science. If you're in science, please stay in science. Face the challenge and you will be surprised at how much power you have. And our practice is that keep some hours for our intellectual nourishment every day. We can do everything out of 24 hours, but we keep 2 hours, no matter which part of the day or night or any time. We need to keep that hour, detach ourselves and nurture our brain and with knowledge, and so that we can do something, contribute. So I like to keep that practice for myself, even though I can be somewhere, but I like to spend some time on my research every day. And we also believe that the more we use our brain and mind, the more beautiful we look. So that's why I think the scientists look quite beautiful. And so ICMMS has been a topic of research project at Georgia Tech, Florida International University, Ohio State University, and other places. And ICMMS featured also at Western Washington University, Voice of America, American Physical Society, Ohio State University Media, Ohio Supercomputer Center, and in various public talks. People, their support also for making this kind of society, so we can do something in a better way, helping and make our advances faster. So to become a member, we just need this information, very simple information, your institutional, educational or professional institution address and your degree, that thing, you need to email that to me. And Professor Hanan Gowda can help you with that. And the membership is free, and it is for lifetime unless you want to quit, just leave the society. And there is a website for it here, and for the ICMMS. So right now we have members from 32 countries, and these are the countries where we have members from. And this is a beautiful picture, I think, from Taiwan University, where I gave a seminar, and the girls were there, lively girls. I wanted to take a picture, but they all went for their abaya, and they put on their abaya, then I took the picture. Okay, so I'd like to say what are the activities we have under ICMMS. Current activities include lectures. Lectures are very effective. You go to a gathering and talk about unity, talk about science, is very effective. So and give support to women. And we do networking. We do extensive networking to enhance each other, give information, involved in scientific collaboration. We also connect for scientific collaboration. We share our success news through in ISM newsletters, and we circulate useful scientific news, also scholarship opportunities and job opportunities. We help with information for higher education in the US, and also even write recommendation letters. We celebrate International Women's Day and also give recognition for excellence. And this shows that 2013 celebration of International Women's Day here at Aligarh Muslim University. And this shows the same celebration, but in 2018. And here she is doing her PhD. She's from Iraq, doing her PhD at Aligarh Muslim University in nanotechnology, and she was nominated to be recognized for her dedication and work. Recently, we also have introduced a magazine, e-magazine. It's called An-Nisa. In An-Nisa, we include the stories of women, not the ones whom we read in the textbook or in Wikipedia or history books. This includes the stories of the extraordinary women who are making the history right now, inventing the path of science through their dedication, intelligence, and truly honest minds. And regardless what the history will do in the future record, but we are all working, making the history right now. So it includes those kind of articles, and we like to keep the same theme for future issues. And you are welcome to submit your success story, a poem or piece of art, for this issue. And just email those to me. This shows the impact of lecture. So in 2014, I'm giving a lecture at the University of Kashmir. It was a snowy night, and with all the snow, a lot of women showed up. It was a full house. Surprisingly, I felt very honored to be with so much response. After 2 years, I was giving a lecture, similar lecture at University of Kashmir, with some science also for this. So this is called at the Women's College on Maulana Azad Road, Degree College. And when someone was introducing me, she told the same things. She quoted the statements that I made, 2 years ago at University of Kashmir. I was so surprised that she was quoting me. So I asked her, where did you get all this statement? She said, well, they are all recorded, whatever you said. I felt honored. And after my talk, a girl from Ladakh came. This is the girl in our picture here. Ladakh is near the China border of India and very isolated, open, beautiful, but not many universities are there. So she came Kashmir for her study. She was so inspired that by the lecture that she came and she wanted to express her gratitude. She asked me whether she could recite the Quran. I said, sure, you do, please. And she recited the Quran to show her happiness. So this shows the other work we do, and the famous scientist of Egypt, Professor Lutfi Al-Nadi. She's a member of our society, and from Ohio State, myself and Cairo University, Professor Lutfi Al-Nadi, we made the memorandum of agreement of collaboration and exchange things, and together we coordinated the whole thing. And here, and it's up at the signing ceremony. They are the deans of engineering and the faculty of sciences. Then she introduced her to Aligarh Muslim University, and where she came in 2016, and she made an MOA with the Aligarh Muslim University. Here, the vice chancellor of Aligarh Muslim University is giving her a recognition with the Indian country symbol. And this shows how we do network helping involvement. She is our member in Morocco at University of Shoaib Dukali University. She wanted to do some research collaboration, but I was not in pure plasma physics like her, so I gave course. So it's a regular course with certificate, and it was completely done under the Innovation Center. He's the director of that Innovation Center, Professor Hamid Nebdi, and he organized everything. But she's the one who initiated and involved people, and I gave the course. 75 people signed up and took the course. This is another session at Wayne State University in Detroit, Michigan. And the chairman, it was a female chairman. She asked that, we want to have that female physicists together. Since you have a society, please, make something together. And then the first time we had a very nice physicists together, all female physicists, and someone sitting here is talking. Her name is Caroline Morgan Pond, and she went to Princeton for her PhD, but when she was younger, even in this country, she was discouraged to pick science. So that's what she was explaining. And she is a Professor Simin Rubab from National Institute of Technology in Srinagar. She wanted to have some connection with the Dhaka University in Bangladesh. So I made the connection. She went there and gave talk and make connections. And this is this picture she sent me standing in front of the physics department of Dhaka University. And she's Munira, whom I met at Dammam University in Saudi Arabia, who wanted to do laser spectroscopy experiment, but was not able to do it. So then I connected her to our another member, Professor Burchin. She has a very nice laser spectroscopy lab in Miami University in Ohio. When I mentioned that, she said, sure, let her come. So she had the scholarship, came, and she did her experiment, and both of them visited Ohio State University and gave talks. So that's how we have been connecting and networking and enhancing each other things. Okay, so I like to mention here, I had also one student who denied marriage in India and applied to US and joined the University of Texas. And And also we have made connection with various other countries whenever it is. I'm just talking about what I'm doing, but other members are also doing the same thing. It is Samor Nahala. She was a young student when I met her, but ultimately she's also a member. Ultimately, I brought her under a program to Ohio State University where she did her part of her PhD. Similar, Soleha also a member did her PhD. She's making presentation. This is her advisor at Ohio State University. And this is Zar from Palestine. Actually, she's from Gaza. She was doing PhD at Cairo University when I was teaching. And she was very devoted. I liked her. I have given her recognition. And I did not know that Palestine has universities. Through her, I came to know a lot more about Palestine, and I founded a program at Islamic University in Gaza, and where they get awards, and you can see that in that award, most of them are females, and the award winners are holding the certificates. And this is another. I got a grant to hold 9 workshops in India for minority, which is largely Muslims, and disadvantaged female students, to encourage them to take STEM discipline, choose STEM disciplines, and come to US for higher degrees. So in those, so I had 9 workshops at various cities, and I implemented the members of ICMMS in India, and she is Professor Noor Zahra. She's in Saudi Arabia right now at Princess Noda University. She organized the workshop for me in Delhi. So that's very helpful. And most of the applications strangely came from Muslim. I was very pleased, the girls, and some of the girls are here from Kashmir too. So this is from for a Delhi workshop. And I put this picture just to say that she is Professor Nazma, Dr. Nazma Heptullah, who got her PhD in biology at age 22. Then she became a professor at age 22. And then she got position at NASA, but didn't go. But she went to the government to serve the government, and she did a lot of developments for Indian technology, electrical side. And but we don't. Nobody mentions her name. We don't know her name. But quietly, that or unquietly, whatever it is, that's what she did. So this is she's an also and model female, I would say. And at the workshop, everyone was so motivated that they didn't know, the participants didn't know how to go for higher education, how to apply to US, and they got motivated very much by our lectures. And so one student wrote, Nabiha, she wrote, "Thank you for availing us the wonderful opportunity to study abroad and opening up the portal for exemplifying my knowledge, which proved to be great help for me to achieve first position among BSc students of my university." See, she's so encouraged. She's not the one. We got letters who did. They feel so encouraged and did so well. And there's another girl, Maimuna Saik also wrote, "I used to top in the Rayalaseema University in MSc Physics entrance exam. I was lucky to be one of the participants. My aim is to be an ideal for others, like Albert Einstein, Abdul Kalam, who is also the father of missile in India. I don't know whether you heard about him, but he is the one who founded the missiles in India, and not least the Sultana ma'am. Okay, now this is about, I just like to say Rala, who became a member of the society when she was a student. After PhD, she got a fellowship. She came to fellowship, US-India fellowship. She came to do a postdoc with me, and she attended conference, and she was very pleased to meet the Nobel laureate whose lecture she used to listen to when she was in India. She wrote a number of papers. She has total 28 publications. When she went back to India, she did not get positions for 3 years, but she did not give up hope. finally she got a position as assistant professor of physics at a government institute. So now I like to say we have some chapters and various places. The Ohio State University has a chapter of ICMMS. It consists of all students only, and I'm the and faculty advisor. There is also a chapter at East Carolina University, USA, chapter at Aligarh Muslim University. Also, there is a chapter in Kashmir, but which includes any female. It is not only NIT Kashmir, but all universities over there. They can be member of ICMMS Kashmir. University of Karachi in Pakistan, NED University of Engineering and Technology in Pakistan, and Islamic University of Gaza in Palestine. And today, inshallah, we'll open up another chapter in Egypt. We can call it ICMMS Misr, if it is all right with everyone. In the name, we'll inaugurate it by the name of Allah. This is a picture, with Professor Hanan Elhouse, and I have proposed her to be the founding president of this ICMMS Misr, and Professor Medhat and Abraham has been very supportive of female education and science and everything. So this is a picture, and I gave a lecture at the National Research Center, and she invited all the females to come to meet me. He's extremely supportive, and for that society, I would like Professor Lutfia Al-Nadi to be an advisor, as I mentioned, and I'm also want to be an advisor, and she will be the founding president, and she can form a committee from other members from you. And I would like to encourage students to be also part of it, serve in the committee, and pray that may Allah bless us with the intellectual power to solve mysteries of His creations and for the benefits of humanity. Amen. Before I finish, I just like to just mention our practices. My time is up. I will just go quickly. That for the sake of dignity and stand, make only positive comments about Islam and Muslims. Never say anything negative. Do not go into debate with others unless you are sure that you can win. So if they say something against Islam, just keep quiet. Don't do anything. Whenever you get opportunity, advertise your own people. That's very important. That will bring respect for Muslims. Regardless of our personal opinion, whether I like that person or not, will always support and help the person if he or she is doing something positive and productive. And if you have a negative point for someone, that person is not good, please don't say it. Be patient and quiet. And there are plenty of people out there who can say bad things about Muslims. So you don't need to say anything to others. And if you have a zero comment, just remain quiet. Do not initiate anything. If you think supporting other groups against your own kind and they will do some justice, it doesn't work. It will end up like digging your own grave. But you should speak out your views forthrightly but politely, and also listen to what others say with respect, even if you disagree. Also, we need to learn how to live with diversity and opposition, but with respect. But we should protect our own identity. If you are the first Muslim making efforts for advances, be ready to absorb insult, put downs, non-cooperative attitudes, and build. But what can you build? Like that is what I do. Build a mental Teflon coating around you, so nothing sticks to it. Whoever say whatever, your brain should not absorb it and put it in your brain and be disturbed. Do not get disturbed. Just wipe it off, just like Teflon coating. And the impact you will, when you do that kind of things, you absorb and go ahead. You will be the pioneer because you have led the way. You observed all the insult and went ahead and become the pioneer. So you're making the way for the people to follow you, to go up and remain united with Muslims and support and promote each other. Okay, that's I want to say. Thank you very much. And I will take the question. And may Allah bless all of you. Assalamu alaikum. To stop recording.
Be presented by Professor Wafat Ismail keynote lecture number 9, titled Zebrafish, the little creation whose contribution is changed by medical research.
Good afternoon, everybody, and thank you for this heavy scientific conference. My talk is about zebrafish. It is a very little creature, only 3 centimeters, and it has many applications in nanomedicine. And it is an alternative model for preclinical investigation of nanomedicine for the last 15 years. It is actually an important creature, widely used vertebrate model organism in scientific research and drug development, preclinical drug development, exploited now on a larger scale by a consortium of European zebrafish groups who generates, this is very important, zebrafish knockout on request. So they are waiting for knockout to investigate and make several projects. Zebrafish has been conducted for 60 years of research. Why? Because they have surprising number of common features with humans, making them a very good animal model organism. They are in vivo model, which means that they are studied in their natural environment. They are cost-effective for drug discovery also. Their embryos are transparent, allowing the observation of the morphological changes that occur during their development. And here are some references about zebrafish and zebrafish welfare. Zebrafish consists of 60 years of UK research. And here are the sites available for the welfare. Actually, although zebrafish have been used in research for 60 years, we can still learn about their preferences. Watching zebrafish in the wild and studying their ecology can help scientists do this. Zebrafish in their native environment in India and in laboratory aquaria. There is a European consortium, and this slide is very important. This is now exploited on a larger scale by a consortium of European zebrafish groups who generate zebrafish knockout on request. They are waiting for request for collaboration. Here are the little amazing creatures, zebrafish in a small aquarium. Also another model of the zebrafish and the other colors. Actually, for 15 years, about 100 articles in 39 journals were most cited between 1990 and 2015. Why use in research, in scientific research? They have fully sequenced genome, easy genetic manipulation, external fertilization and rapid development, nearly transparent embryo. They are small and robust, and they are cheaper to maintain than mice. Why zebrafish are used in research as a model organism? It is considered as a model organism because they have surprising number of common features with human, making them a good animal model organism. They are an in vivo model, which means they are studied in their natural environment. They are cost effective also for drug development. Their embryos are transparent, which allows observation of any morphological changes that occur during their development. Zebrafish are used in research. Why? In drugs, preclinical. Zebrafish is an important and widely used vertebrate model organism in scientific research, for example, in drug development, in particular preclinical development. This is about the international cooperation concerning zebrafish. We can see that the United States are dominating, followed by Japan and France, and Germany also are playing with this treaty. The top 100 cited articles network about zebrafish, we can find that they're covering different disciplines, just like biocompatibility, bone morphogenetic protein, and other blood research, cranial and other classifications. Actually, they said that the zebrafish formed a splash in FDA research. Here is the control, and here is the ketamine, only 0.5 millimeter, and this is 2 millimeter ketamine. Here are the comparison between the larva and the adult in centimeter. These are 3 millimeter, but they are complete. All the organs available, and this is in 3 centimeter. How 3 R's are applied to zebrafish? What is the 3 R's? Replacement, reduction, and refinement. For the replacement, larval zebrafish could be used to replace some animal toxicity. Studies relevant model for the system target gene pathway, mechanism, tissue organ, etc. with validation studies. Reduction, the second R, model for toxicity, larva used to identify toxic drug candidates, safer molecules to be tested in mammalian models. The third R is refinement, because the embryos are fertilized externally and are transparent through early days. Drug development model. The zebrafish is an important widely used vertebrate model organism in drug development, in particular preclinical development. It is also notable for its regenerative abilities. The third one is it has been modified by researchers to produce many transgenic strains. Major role in hematopoiesis and blood as human. They have the same sequential multilineage hematopoiesis process as humans with the same distinct lineage. Therefore, provided scientists with many insights in both blood lineage development, pathways, and blood disorder. China zebrafish resource center. Actually, there are various transgenic and mutant strains were stored at China zebrafish resource center. This is a very big center. A non-profit organization which was jointly supported by 2 important two important facilities, Ministry of Science and Technology in China and the Chinese Academy of Sciences. Pharmaceuticals and the drug effects. The internal drug exposure is essential. The pharmacological effect, translating experimental results from zebrafish to higher vertebrate like humans, requires concentration effect relationship, and it can be derived from pharmacokinetic and analysis. To date, only a single pharmacokinetic model was performed for paracetamol in zebrafish. Several transgenic models, melanoma, leukemia, pancreatic cancer, hepatocellular carcinoma, blood clotting, blood vessel development, heart and kidney diseases. Vertebrate innate immunity. Actually, we have two immunity, innate immunity and the adaptive immunity. The innate immunity is a system capable of phagocytic activity by 28 to 30 hours post-fertilization. The adaptive immunity is not functionally mature until at least 4 weeks post-fertilization. For infectious diseases models, for a wide range of bacterial, viral, and parasitic pathogens, tuberculosis provides fundamental insights into the mechanism of pathogens and mycobacteria. Neurons in the retina, glaucoma, and diabetes-related blindness. This is very important actually because we have a big number of diabetes patients in Egypt. Robotic technology has been developed for high-throughput antimicrobial drug screening using zebrafish infection model. Zebrafish retina characteristics has also extrapolated at University College of London, grew a type of zebrafish adult stem cell found in the eyes of the fish and mammals that develop into neurons and in the retina. These could be injected into the eye of the treated disease that damage retinal neurons. Nearly every disease of the eye, including macular degeneration. It is a model model organisms in drugs and in bone. The ability of zebrafish embryo to absorb the chemicals and testing new drugs against muscular dystrophy. Bone physiology and pathology as a model organism for bone metabolism, tissue turnover, and resorbing activity. Bone physiology and pathology model. Actually, bone metabolism, tissue turnover, and resorbing activity. Study osteogenesis, which is the bone formation, evaluating differentiation, matrix, deposition, activity, and the crosstalk of skeletal cells to create and isolate mutants, modeling human bone disease. Testing new chemical compounds for the ability to revert bone defects. The larva to follow new de novo osteoblast formation during bone development. They start mineralizing bone elements only 4 days post-fertilization. Adult zebrafish to study the age-related bone diseases. We know, of course, about osteoporosis and osteogenesis imperfecta. Scales, القشور يعني بتاعت السمك. Scales, osteoblast, bone building, osteoclast, bone eating, bone remodeling, and metabolism. The elasmoid scales of zebrafish function as a protective external layer are little bony plates made by osteoclasts, bone building. The scales also act as main calcium storage of the fish, cultured ex vivo, kept alive outside of the organism in a multi-well plate. Manipulation with drugs and even screening of new drugs that could change bone metabolism between osteoblasts and osteoclasts. Model bone physiology and pathology. Bone metabolism, tissue turnover, and resorbing activity are evolutionarily concerned. Osteogenesis, which is the bone formation, evaluating differentiation, matrix deposition, activity, crosstalks of skeletal cells to create and isolate the mutants, modeling human bone disease. Testing new chemical compounds to revert bone defects. In the larva, to follow de novo osteoblast, new formation of osteoblast formation during bone development, they start mineralizing bone elements only 4 days post-fertilization. Adults to study complex age-related bone diseases such as osteoporosis and osteogenesis imperfecta. Osteoporosis and osteogenesis imperfecta, these are age-related diseases. Adult zebrafish to study complex age-related bone diseases as osteoporosis, osteogenesis imperfecta. The scales also act as the main calcium storage of the fish. The elasmoid scales of the zebrafish function as a protective external layer and little bony plates made by osteoblasts in bone building. These exoskeletal structures are formed by bone matrix depositing osteoblasts building and remodeled by osteoclasts. Drug manipulation and screening for new drugs. New drugs for bone metabolism. The adult zebrafish are being used to study osteoporosis and osteogenesis imperfecta. The scales in a elasmoid function as a protective external layer which are made by osteoblasts and are remodeled by osteoclasts. The scales are the main calcium storage of the fish, allowing to function. Manipulation with drugs, they can be cultured ex vivo, kept alive outside the organism in a multi-well plate. Screening for new drugs that could change bone metabolism between osteoblasts and osteoclasts. Genetic obesity and overnutrition. Obese zebrafish, dysregulation and of lipid controlling metabolic pathways leads to weight gain without normal lipid metabolism. Good models for studying obesity in humans store excess lipids in a vesicular, intramuscular, and subcutaneous adipose deposits. Genetic obesity is usually studied in transgenic and mutated zebrafish with obese genetic genes. Diet-induced obesity, high-fat diets, and general overfeeding diets both show rapid increase in adipose deposition, increased bone morphogenetic hepatosteatosis, and hypertriglyceridemia. Pancreas development homologous to mammals as mice diabetes. The signal is a signaling mechanism and way the pancreas functions are very similar with human. Pancreas has an endocrine compartment which contains a variety of cells, pancreatic cells, polypeptides, beta cells, insulin, glucose tolerance, tests zebrafish diabetes in human, exactly like human. Insulin functions are also being tested in zebrafish, which will further contribute to human medicine. Zebrafish Husbandry Association. This association is very important and they are opening doors for collaboration. Zebrafish Husbandry Association is a non-profit organization developed to promoting and developing zebrafish husbandry, standards through education, collaboration, and publication. The zebrafish information network is a database of genetic and genomic data of zebrafish Danio, this is its name, as a model organism. As a model for human biomedicine, several groups have investigated zebrafish as a model for human biomedicine, including molecular mechanism of skin, neurological disease, muscular dystrophy, cardiac muscle regeneration, eye disorder, acute renal failure, hematopoiesis, immunological diseases, cancer, and autophagy. Why zebrafish are used in research? They have common features with human, making them a good animal model organism because they are an in vivo model being studied in their natural environment, zebrafish journal. They are cost-effective for drug discovery. Embryos are transparent, which allows the observation of morphological changes occurring during development. This is the site for models of the zebrafish in Korea. It has wide scales of zebrafish function as a protective external layer, and there are little bony plates made by osteoblasts. These exoskeletons structures are formed by bone matrix depositing osteoblasts and are remodeled by osteoclasts when eaten. Also act as the main calcium storage of the fish, and it can be captured ex vivo, kept alive outside the organism in a multi-well. Drugs allow manipulation with drugs and even screening for new drugs that could change bone metabolism between osteoblast and osteoclast. Bone physiology and pathology. Model organisms for bone metabolism and tissue turnover, resolving activity. These processes are largely evolutionary. Conserved start mineralizing bone elements as early as only 4 days post-fertilization. To study osteogenesis, which is the bone formation, evaluating differentiation, matrix deposition activity, and to stop of skeletal cells to create and isolate mutants, and modeling human bone disease, and testing new chemical compounds for the ability to revert bone defects. The larva, to follow new or de novo osteoblast formation during bone development, and for the other, they study complex age-related bone diseases, just like us exactly. We have osteoporosis and osteogenesis imperfect. The anosmoid scales, these function as protective external layer, and the little bony plates made by the osteoclasts. These exoskeleton structures are formed by one matrix depositing osteoblasts and remodeled by osteoclasts. The scales also act as the main calcium storage of the fish. They can be captured ex vivo outside, kept alive outside the organism in a multi-well plate. This allows the manipulation of drugs and screening for new drugs that could change bone metabolism between osteoblast and osteoclast. Chemical research in toxicology. The 3 Rs. 3 Rs, replacement, zebrafish assay using larval zebrafish, replacement, some animal toxicity studies. First established that the larval zebrafish is a relevant model for system, target gene pathways, mechanism, tissue organ with validation studies. Reduction as a first-tier model for toxicity used to identify toxic drug candidates, allowing safer molecules to be tested in mammalian models. Refinement to animal study design. Their embryos are fertilized externally and they are transparent. Refined embryonic and larval fish model offers refinement to animal study. embryos are transferred, fertilized externally, allowing non-invasive observation of toxicity and recovery. 2 stages, approximately 5 days post-fertilization for the larva and 3 months for the adult. In body volume, the larval fish is easy to work, times smaller than the adult. Hundreds of laboratory animals, chemical research, toxicology. This is released very recent, only 2 years ago research. Infectious disease research. Zebrafish are susceptible to strain Streptococcus, used as a model for one of the most frequent pathogens in human, Streptococcus pyogenes. Astronauts develop osteoporosis in the microgravity encounter during the space mission. That's why they send the fishes to the space. European Space Agency ESA supports a project using a transgenic fish model to study gene expression profiles on Earth compared with those in microgravity environment. ESA informed consortium, and this is involved in Germany. This is its site on HTTP. Actually, 60 years of research and preference. North Indian zebrafish are seen in their native environment as well as in laboratory aquarium. Here are some references on the observation of wild zebrafish in India. Zebrafish journal, there is a journal for zebrafish, Stephen C.H. and this is very new, 2021. Enrichment for Sunday morning, final day, circle on 2019 on the observation of wild zebrafish in India for the zebrafish journal. The second most used animal in British research. Fish specifically. Zebrafish are used steadily, increasing over the last 2 decades. Fish account for approximately 50% of all the British research. For detailed animal research statistics here, www.animalresearch.info on fish and zebrafish. RSPCA has issued guidance on housing and the care of zebrafish for science. Actually, these are 10 points working with zebrafish in England. The most important is zebrafish embryo are nearly transparent, which means that they can be observed embryo with a microscope. Actually, these 10 points are very important and they are performed by the British research. Here, the models, the WWZ model.org CO, this is now exploited on large scale. Consortium of European zebrafish groups who generate zebrafish knockout on the request. Yeah, they are waiting for a request from other parts of the world. ZIRC is a Zebrafish International Resource Center. They have a responsibility with 29 to 50 available for distribution for research community. The radio, this is its name, is also one of the few fish species to have been sent to space. They sent it to space to find what is going there. Research with the radio has yielded advances in the fields of developmental biology, oncology, toxicology, reproductive studies, tetrachology, genetic, neurology, environmental sciences, and muscular dystrophy evolutionary theory. Zebrafish journals, Stevens, these are 2 references. They are available for scientists. Thank you.
Professor Albert, for this very informative and interesting presentation.
Thank you.
We can now open for discussion or some questions.